Good afternoon, everyone. Welcome back, and let's do and continue our course on science and technology. Today is class number 5. What we are going to read is AEFI. AEFI stands for Adverse Effects Following Immunization. Adverse effects are basically side effects. So there is no such thing as a perfect vaccine. कोई vaccine perfect नहीं है, because if you what would be a perfect vaccine कि आपको 100% disease से protect कर ले और कोई भी side effect भी ना हो. This does not exist in the world. When you take a vaccine, what you are doing is you are substituting a mild illness so that later you don't get a severe illness. So this mild illness will happen if you take a vaccine, right? Some or the other side effect, like if you take vaccine, you might get fever, you might get a scab, you might get a scar. I will ask you to take, if you're wearing a t-shirt, just move the left sleeve up and look here. All of you will see a white scab or a white scar, a part of smooth skin. This must have happened when you were given the tuberculosis vaccine called BCG, which we will study today. When you were given this TB vaccine, here you will have a white scar. It can be here or here, depending on which state you were given the vaccine. Mostly it's on the left side. So you will see a small scab here. This will be because you were given the TB vaccine and there was a small side effect of that vaccine, right? So all vaccines will have some or the other side effects, right? Because you are trying to give your body a mild infection to protect from later a very big infection. Now, effective vaccines that are producing the principle of memory, that are producing protective immunity, are those that give you very little side effects and give you a lot of immunity, a lot of memory. So what would be an effective vaccine? An effective vaccine would be that I give you a little bit of a pathogen with its antigen. I take the antigen, your body recognizes and forms memory. The side effects are very little. The memory generated is very much. Side effects are very little. Memory generated is very high. So when you will get the actual disease, you will already have memory and your response will be much quicker. So an effective vaccine is that has less side effects and more immunogenicity. What is immunogenicity? Word breakdown. Immunogenicity, the ability to generate immunity. And which immunity? Acquired immunity, memory-based immunity. Immunogenetic vaccine, immunogenicity should be high, side effects should be low. But it is not possible that you will have 100% immunogenicity and 0% side effects. This is not possible in any vaccine. Some or the other side effect will always be there. Now, the adverse effects. Adverse effects means some real bad thing that happens to you after taking the vaccine. Let's say someone gets a brain stroke after a vaccine, or someone dies after a vaccine. Due to the clinical trials that are taking place in vaccines, this thing generally does not happen. But in case it happens, it's called a AEFI and it has to be reported to the government. Now, why do AEFI happen? They can either happen due to the vaccine, right? That the product or the quality of the vaccine is defective. Or they can happen due to the vaccination process, that the person who was giving you the vaccine did not know how this vaccine is supposed to be given. Or they can occur independently from vaccine, meaning they can occur regardless of the fact that the vaccine was good quality and it was given properly. It's just something that was seen coincidentally. So there are three types: due to the vaccine, due to administration, and independently from the vaccine. What can happen? You can have vaccine product related reaction, vaccine quality defect related, immunization error related. These are the types of AEFI. This is just to explain you that vaccines will have some side effects and some can have adverse effects. In this case, what we do, we report to the government that this effect has happened, and then the surveillance starts. That okay, is this a small one-time incident or is there a problem in this vaccine? What are the types of vaccines? Types of vaccines can be on the basis of how the vaccine is manufactured or how the vaccine is given to you. These are the two ways. So on the basis of administration, depending on how the vaccine is given to you, they can be one IM. IM stands for intramuscular. These vaccines are given to you in the muscle. Then they can be IV. IV means intravenous. These are given to you directly in the veins. If you have gone to the doctor for vaccination, some vaccines would have been given to you on your bum, right? Those vaccines are intramuscular because your bum is one of the largest muscles in the body. Some vaccines must have been given to you here because this cubital vein is the easiest way to inject a vaccine, right? Some must have been given to you on either your skin or on the fat layer. They are called subcutaneous. SC means subcutaneous. Now cutaneous means skin. So subcutaneous means just below the skin. So subcutaneous, intramuscular, intravenous. Through the name it's clear. Otherwise there are some nasal vaccines that you just have to inhale. And the most common vaccine that Indians get is an oral vaccine called polio. The polio vaccine is famous for Do Boon Zindagi Ke. That is an oral vaccine. You are just drinking the 2 drops and it is working like a vaccine. Okay. Now, this was the type of vaccine based on how it's given to you. Now there is a type of vaccine based on how the vaccine is made. What is the principle of vaccine? I'll just repeat again so that the next part is clear. I have to give you the antigen. I hope everybody has revised, so this is not a problem. I have to give you the antigen so that your B cells generate antibodies, so that your B cells start having memory. Okay, I do not need to give you the pathogen. The entire pathogen is not needed. Only the antigen is needed. So basis of vaccine formation is also on the basis of how the antigen delivery is happening. How am I delivering the antigen to you? In what form? In what way? Because I don't want to give you the pathogen. If I give you the pathogen, you will have disease. So I want to give you either a killed pathogen or a weakened pathogen or a modified pathogen so that you don't have disease, but you still get the antigen and you generate antibodies and generate memory. Now there are 3 basic approaches. One is I use the whole virus or bacterium. I use the whole virus or the whole bacteria. Second is I use the genetic material, and third is I use only the parts that are antigens. I'll show you all 3 of these approaches in detail. These are the types. I'm not reading them here because we are going to do them all in detail. So the first type of vaccine is inactivated vaccine. What am I doing? This is a pathogen. It has certain antigens. Now, what do I want? I want that my B cells get the idea that they are able to see this antigen. They are able to see this antigen and generate memory, but the pathogen should not be powerful enough to cause disease. Because अगर मैंने pathogen ही दे दिया, then what's the point? I only have to give the antigen without the pathogen to cause vaccination and memory generation. So what I will do is, through some technical manipulation, I will inactivate this. I will modify this bacteria that causes disease. I will kill the bacteria. Okay, let's say I kill the bacteria. Now the bacteria cannot cause disease, but it still has antigen. I will give you the killed bacteria. Your body will see the antigen on the killed bacteria. Your body will generate memory. So inactivated vaccines are using killed version of the germ that cause a disease. I hope it's clear that you are killing the pathogen, but still the antigen is there on the outside. So the antigen gets delivered and you are generating memory from the antigen. Now, inactivated vaccines don't provide immunity that's as strong as live vaccines. What are live we'll see. But if you kill the bacteria, the immunity generated is not that great. It's decent, but not that great because immunogenicity is low. So in this case, you will need booster doses. So this is why you must have heard the term, अब तो सबने सुन ही लिया है COVID के term में booster, but other vaccines also have boosters, right? So in case of inactivated vaccines, because the microbe is killed, you will need more booster doses. This is why inactivated vaccines need booster doses. I am killing the pathogen, just giving the antigen. Now, inactivated vaccines are used to protect against which diseases? Hepatitis A, flu, polio, rabies. These vaccines are inactivated vaccines. This list I will ask you to remember because sometimes it's asked. Hepatitis A, flu, polio and rabies have inactivated vaccines. Okay. Now, the second type is live attenuated vaccine. Live attenuated vaccine. Attenuation means weak. Attenuation means weak. So what we need to do? We need to word breakdown. In this case, you are being given a microbe. In case of live attenuated vaccines, you are being given a microbe that is weak. Its ability to cause a disease is much lesser than what a normal microbe would do. So what will happen in this case? In this case, your B cell will come, it will look at the antigen and it will generate memory. B cell does not care what is inside. But since it is a weak pathogen, it will not cause disease. You will get very mild disease, very less disease, right? In this case, you will get fever. Maybe one day you will get fever. होगा आपने कभी ना कभी वैक्सीन लगाया, एक दिन तक फीवर रहता है. This is usually that case. The pathogen is weak, the antigen is as such, memory is generated, and then the response is much stronger. So in this case, I am weakening the microbe. I am not killing it. So the microbe is still live, but it is weakened or attenuated. That is why the term live attenuated vaccine. Live attenuated vaccine contains the weakened or attenuated form of germ. Now, why are these vaccines good? They are similar to the natural infection. जैसे जब natural infection होती तो भी live vaccine, live pathogen ही आता, but the live pathogen would have been much stronger. Here the live pathogen is much weaker, but there is a lot of similarity, right? So they are similar to the natural infection and they generate a long-lasting response. Because they are similar, very close to the natural process, their immune response is much more long-lasting. And that is why these live attenuated vaccines require either no boosters or less boosters. Less boosters are needed because their immune generating ability is much more. Okay. Now, forget the slide. I want you to think of a disadvantage of live attenuated vaccine. Think about it. There are many disadvantages, but I want you to come up with at least one. What could be a disadvantage of a live attenuated vaccine? I am repeating, it is very close to the natural infection. What is a disadvantage? Think about it. Best answer is that there are some people whose immunity is very weak. Right? Their immunity is very weak. If your immunity is very weak, even if you are given a live but attenuated vaccine, you will not be able to tolerate it. Okay? You will not be able to tolerate it if your immune system is already very weak. It does not have the ability to even fight the weak pathogen. Then you will have a problem with live attenuated vaccines. And this happens in cancer patients. ये आंसर कई लोगों ने दिया है, good. This happens in diabetic patients. Okay. So in diabetic and cancer patients, this is a common occurrence. Alright. So live attenuated vaccines are not good for those people who have a compromised immune system. Second thing, you are keeping a microbe alive. Although it is weak, but you are keeping it alive. You are not killing it. To keep it alive, certain conditions have to be maintained. So these require better refrigeration, better cooling capacity. Okay. So in live attenuated vaccines, there are two disadvantages. One, if you are weak in your immune system, you will have a problem. Second, if you are keeping it, you will have to keep it in a cool condition. Right? So these vaccines are used against MMR. MMR is a combined vaccine against measles, mumps and rubella. Then rotavirus vaccine, smallpox vaccine, chickenpox vaccine, yellow fever vaccine. Again, this list is in the slides and you can see from here. I am not asking you to print the slides every day. You can see them digitally and add to the one pager. That is why the one pager was made. Okay. All right. अब आते हैं next चीज पे. What we were doing so far is we were taking this pathogen, it has certain antigens, right? Either I was killing it or I was weakening it. So then scientists thought, why not we come up with a different approach, because our molecule of interest is only this one. Okay, we don't care about that. And this was a question by somebody, beautiful, insightful question, that why don't we give only antigens? Why are we giving this pathogen alongside? We are killing or weakening it, but if we can get this out and I can just give the antigen, that will work. Earlier, this technology was not available. That is why many vaccines have been formed like this. But now that we are able to do this, we are able to extract the antigen, we are giving only antigen as vaccine. So what will be the advantage of such a vaccine? Not no side effects. I have told you there is no perfect vaccine, right? Not no side effects, less side effects. And they can also be tolerated by those whose immunity is weak. Even they can tolerate this, right? So this is what is called a subunit vaccine. A subunit vaccine is that contains only the purified parts of pathogen that are antigenic. The parts of pathogen that are antigenic, the antigen of the pathogen, only that is purified and delivered. The entire pathogen is not delivered. Okay, this does not contain the whole pathogen. It contains only the antigenic part, only one small part, only one small subunit of the antigen of the pathogen is there. That subunit is antigen. So of the entire pathogen, it does not have the complete pathogen. It only has a subunit of the pathogen, which is the antigen, and this is why they are called subunit vaccines. Now they do not contain any live components, so they are suitable for people who are immunocompromised. Suitable for people who are immunocompromised. Again, word breakdown: immunocompromised. Immunocompromised means compromised immunity, don't have good immunity. The other term we had studied is immunogenicity. Immunogenicity is the ability of the vaccine to generate an immune response. So immunocompromised, immunogenicity. These are two different terms. In immunocompromised people, subunit vaccines are a very good idea, but they are far away from the natural infection. The natural infection does not happen like this, so their immunogenicity is less. I hope this is clear. Okay. Now we are going to see recombinant vector vaccine. Okay, this is a very interesting vaccine, and let's see how it is made. Recombinant, this term we have studied before. Recombinant stands for recombinant DNA technology. We have heard this before, where we are joining two pieces of DNA that are generally not seen together. Joining two pieces of DNA generally not seen together is called recombinant. What is a vector? Vector means carrier. We have seen plasmids were being used as vectors, right? So vector is carrier, recombinant is DNA combined. Recombinant vector vaccine. Again, word breakdown. So let's understand what we are doing here. In this case, what I am doing is I am taking either a bacterial plasmid, I hope you now know, or I am taking a virus. Now what does the virus generally do? Virus has certain genes. It infects my body using those genes. Okay. Virus has either RNA or DNA, this we are going to see tomorrow, but virus infects my body like we had seen in the HIV virus. It was coming to my body and integrating its DNA with my DNA using the enzyme reverse transcriptase and integrase, like we saw in the video. So what I can do is I can either do genetic editing of this plasmid, remove the gene that causes disease and insert the gene that I want, or I can do genetic editing of virus. I can remove the part that causes disease, I can insert the part that I want. So I am using what? One, I am using a carrier called vector. I am using a vector, either a plasmid or a virus. So I am using a vector. I am joining the DNA that I want. Which DNA is this or which RNA is this? We will just see. I am joining the DNA that I want into the plasmid or into the virus, and I am removing from the virus or the plasmid the part of DNA that it has that causes disease. So I am doing some recombination. So there is recombinant vector vaccine. Okay, which DNA I am joining? This we will just see. Okay, so it uses a safe virus or bacteria to deliver specific subparts of the pathogen. How is it done? We will just see. Right, it is using a safe virus or bacteria to deliver specific subparts of pathogen. This, either we were giving these subparts, this subpart is what? Antigen. Either we were purifying and giving this subpart as subunit, or I am using virus or bacteria to deliver this subpart, right? So immune response is generating without causing a disease. Obviously, since I am not giving the whole pathogen, disease is not going to happen. I am giving only a subpart. Earlier case, I was giving subpart directly. This case, I am using virus or bacteria to give that subpart. How? We will just see. Genetic instructions of making such a protein are inserted into safe virus through rDNA. We will just see this. Okay, but first we need to see what vectors are used. Which viruses are there that are used as vectors? The most common viruses are adenoviruses. What are adenoviruses? We will see tomorrow. But adenoviruses are the most common viruses that are used as vectors. Okay, and sometimes bacterial plasmids are used. The name of other viruses that are used, like vesicular stomatitis virus, measles virus, these are written in the slide. Just read them once. So here I am using a vector like a bacteria or a virus to deliver some part of the pathogen called the antigen using recombinant DNA technology. अब इसको समझते हैं. Let's study this further, then we'll come to the other vaccines. Okay, I'll just make a new diagram. Fine. So let's say that I have a pathogen A. Listen to this carefully. I have a pathogen A. Pathogen A looks like this. It has an antigen called A. Okay, with me so far? There is a pathogen called A, it has an antigen called A. This pathogen must have some part of its DNA. Some part of its DNA must be coding for this antigen, because antigens are usually 99% of the time antigens are proteins. So in this pathogen, some part of DNA, let's say this part of DNA is that gene that codes for this antigen. Okay, now what I want to do is I want to deliver only this antigen, but I found that in this pathogen it was not possible to do the purification and to get the antigen out. नहीं हो रहा था, the technology was not available. Then I found कि इस pathogen में एक gene है that is coding for this proteins, because genes are those part of DNA that code for proteins. Now what I did, I used a technique called genetic editing, और कौन सा molecule और कैसे ये होता है वो भी हम आज ही देखेंगे. I got this gene out. I got this gene out. So I got something like this. This much DNA I got. This DNA codes for an antigen A. Now what did I do? I secondly took a virus called adenovirus. Adenovirus had looked something like this. Okay, adenovirus had its own DNA. Okay, this is just an example. Adenovirus had its own DNA. Now in adenovirus, there was a specific part of DNA that used to cause a disease. Let's say this part here was the one that used to cause a disease in humans. Right? So normally what would happen? The adenovirus would go, it would infect all the cells. Now it will integrate this part of its gene with my cells and cause a disease. This is what is normally happening, because it does not have its own cellular machinery. So it will use my cellular machinery, use this part of its gene to generate a protein. This protein could be an enzyme, and this enzyme used to cause disease in me. Okay, so this is what was normally happening. Now I use the technique of genetic editing. I cut this part. Okay, I can do this using restriction endonuclease. I cut this specific part and got this disease causing gene out. Then I took this specific part and put this here. So I put the antigenic protein into the virus. Okay, so ये हो गया ना recombination. Recombination has happened. I have a vector and recombination has happened. I have put the antigenic gene inside the virus. Now the viral DNA looks something like this. Now the viral DNA looks something like this. Clear? Viral DNA looks like this now. Okay. Now what did I do? I took this viral DNA that looks like this. It does not have any disease causing genes anymore. It only has antigenic genes anymore, right? So it has antigenic genes. It does not have any disease causing gene. This recombinant DNA molecule is put back inside the adenovirus. Now this is what the adenovirus looks like. It is a vector, a carrier of a recombinant DNA molecule. Now I will cause this adenovirus to infect me. That okay, now you infect me. Now when this adenovirus goes into my cells, what is going to happen? Normally the adenovirus does a simple thing. It integrates its DNA into my cells, and when it is integrated into my cells, my cellular machinery produces the proteins the adenovirus DNA usually codes for. So now it will go into my cell. Now in my DNA, let's say my DNA looks like this. Okay, I'll draw the whole thing. My DNA looks like this normally, blue color. The adenovirus comes, it has the ability to integrate. So what it does is, my DNA now starts looking something like this. Adenovirus DNA gets integrated. Okay, now my DNA looks something like this because adenovirus has integrated into my cell. So now when adenovirus has integrated into my cell, now when my body does transcription, then translation, what will it code for? My body will start coding for, originally the normal proteins that are there, they will be coded, my normal genes will be coded for. Then it will start transcripting and translating the adenovirus DNA. In the adenovirus DNA, there is the antigenic DNA. So my body will start now producing these antigenic proteins by themselves. Okay. My body's cellular machinery, it normally codes and understands this DNA, then through the process of transcription and translation makes proteins. So now it will again do the same thing. Look at this antigenic DNA and code the antigen. So what is going to happen? My body's cell is going to look something like this. It will have a nucleus, it will have mitochondria, it will have endoplasmic reticulum, it will have Golgi bodies, but after the transcription and translation has happened, it will start having something called antigenic proteins here. Or I'll draw it the way we were drawing it earlier. This antigenic protein is now there inside my cell. Why? Because it has been coded for. It was coded for in that DNA. That DNA got integrated. Then after this integration, the process of transcription and translation took place. After transcription and translation took place, my body read this code and produced the protein. This protein was the original antigen here. This original antigen has now finally gotten inside my cell. Now, in the process of normal life, this cell will die sometime. or there will be a process through which this antigen will find its way outside. Okay, either this cell will die sometime. Okay, let's just consider the first process, that this cell normally will die at some stage of life. This cell will die. When this cell dies, and this is happening normally, every cell has a lifespan, our body has the ability to produce new cells. So when this cell dies, its plasma membrane or cell membrane will burst open. This antigen then will get released into my blood. Now the antigen has finally reached my blood. And this is what the goal of vaccination is, to get the antigen to the blood. Because as soon as the antigen gets to the blood, what is going to happen? There is a person here called a B cell who is waiting for this. It will look at it, start producing antibodies, generate memory. So I have delivered the antigen. The idea is to deliver the antigen to the blood without the pathogen. This is the basic idea of vaccine. I have to deliver antigen to the cell without the pathogen. So I could not get the antigen separated myself. So I took the gene that was coding for it, used recombination, got it inside a vector called a virus. Then I used the virus to infect my body. Then my body itself started coding for the antigen. Then when that cell died, antigen started getting produced, and then B cell generated memory. Now you might be thinking that this might lead to autoimmunity, right? It will not lead to autoimmunity, and I'll tell you why. Let's say my body has 100 cells. If my body has 100 cells and I get them infected by the virus, let's say only 10 get infected. So these 10 will start showing these antigens. The B cell will kill these 10, but my body has the ability to regenerate this 10. Autoimmunity will happen when my B cell starts to target all 100. Here it will only target those that have been infected by this adenovirus. So I have used recombination, I have used a vector, and I have formed a vaccine. This is recombinant vector vaccine using DNA. Okay, so I hope the process is clear to you. So here we were seeing recombinant vector vaccine. In this case, we are using a safe virus or bacteria to deliver specific subparts of a pathogen. Thing is clear? How have I made the virus safe? Through removing the gene. Or bacteria, I can use the bacterial plasmid also. Same thing. Safe virus or bacteria to deliver specific subpart of pathogen. Immune response is generated without causing a disease. Of course, genetic instructions of making such a protein are inserted into safe virus through recombinant DNA technology. Clear? What are the viruses that we can use? Vesicular stomatitis virus, measles virus, adenovirus. These are commonly used. Viral vector produce immunity from both B cells and T cells. Now this is a new concept, that viral vector vaccines have the ability to produce immunity from both B and T cells. How? Not important for you. Okay, I can tell you if you want, but it's not important. Viral vector vaccines produce immunity from both B and T cells. So now we have understood recombinant viral vector or recombinant bacterial vector vaccine. Now we are going to see a conjugate vaccine. Now what does the word conjugation mean? Conjugation word means combining two things together. Conjugation means 1 plus 1, like if I join two things together, it is called conjugate. So in conjugate vaccine, what are we doing? A conjugate vaccine is a type of subunit vaccine. It is like a subunit vaccine only, but slightly different. Here what we do, we combine a weak antigen with a strong antigen as a carrier. Okay. So what is happening here, that there is one weak antigen. This is an antigen. Okay, I'll use a different color. This is an antigen, but this antigen, if I give itself as a subunit, it is weak. It does not have good immunogenicity. It does not have the ability to generate immunity. It is weak, right? It does generate immunity, but not a lot, not as much as I would like, right? So what I did, I combined it with a strong antigen. This is a strong antigen. When I give this as a subunit vaccine, this antigen, it generates a lot of immunity. So what I did, I combined these two together. I made something like this in one. I combined them in one. Now what will happen when my B cell comes? It will generate memory to both together. How this happens is not important, but if we combine these two things together, we will be able to generate strong immunogenicity, or we will have strong immunity even for this weak antigen. This is how we are doing conjugate vaccines. So clearly they are a type of subunit vaccine. We are taking a subunit only, but this antigen usually did not generate, when I injected it and I saw the trials in patients, it did not generate enough immunity. So I combined it with some other antigen that was strong. Now the B cells will start recognizing both. Okay. This is a conjugate vaccine. Now most vaccines contain a single antigen. Most vaccines contain a single antigen that the body will recognize. But some antigens do not elicit a strong response. Okay. Some antigens do not elicit a small response. So in this case, conjugate vaccine is used in order to invoke an immune system response against the weak antigen. Here I am combining the strong and the weak antigen as a conjugate, as a combination, so that my body gets confused. It does not recognize which is the weak one, which is the strong one. It starts thinking that both are the strong one, and it starts generating immunity and memory. Okay. Now this line is important here. Most likely or most commonly, the weak antigen is a polysaccharide. What that is, let's see in the next slide. And attached to a strong protein antigen. Okay. Most commonly, the weak antigen is a polysaccharide. What is a polysaccharide? I'll just tell you. Is attached to the strong protein antigen. Okay. Now, I was going to tell you because next thing is this only. Most antigens are what? Most antigens are proteins. Most antigens are proteins, and our body usually has good ability to generate immunity against proteins. So most antigens are strong because they are proteins, and our body has good ability to recognize and generate immunity against protein. But some antigens in some bacteria, what happens is, or in some viruses, let's say in some bacteria, what happens is that whatever we are able to get from here, because this is happening at a very microscopic scale. It is not, okay, it's in my hand that this is the bacteria, nikal lo jo antigen nikalna hai. It is happening at very microscopic scale. Very, very microscopic scale. There are probably 10,000 bacteria on my hand right now, and on yours too, and maybe 1 million bacteria on this board, but we are not able to see anything, right? So at this very microscopic scale, we are not able to extract the protein sometimes. Okay, the protein cannot be extracted, but we are able to extract carbohydrate. I hope you know what this term means. So sometimes we are only able to extract carbohydrates from the bacteria. So these carbohydrates are also known as polysaccharides, because saccharin is a constituting molecule of a carbohydrate. So polysaccharide, many saccharin together makes carbohydrates. In carbohydrates, when the immune system sees them, the response is not that strong. So some antigens therefore are weak antigens because we are unable to extract the protein. We are only able to get the carbohydrate. The carbohydrate has weak immune response in our body. But when the actual microbe comes, it will have protein also. But हम वो protein निकाल नहीं पा रहे हैं. तो हमने क्या किया? हमने कार्बोहाइड्रेट निकाल लिया कि चलो कुछ तो निकला, वीक इम्यूनिटी आ जाए. And then to trick the immune system, I combined this polysaccharide that I was able to extract with another antigen that was strong, that was protein based. When I combined them together, the body thought that this is a protein only and started generating strong response. This is what conjugate vaccine was. Now polysaccharide vaccines are polycarbohydrates. They are carbohydrate based. They generate immune response. Mostly it is weak, sometimes it can be strong. But why are polysaccharide vaccines special? Polysaccharide vaccines are special because most vaccines have protein antigens. Only polysaccharide vaccines have carbohydrate antigen. So they are a special type. Okay. Now there is nucleic acid vaccine. You have read two nucleic acid. One is deoxyribonucleic acid. Second is ribonucleic acid. We are using the genetic material from a pathogen to stimulate an immune response. A genetic material from a pathogen to stimulate an immune response. Now this could be done using DNA. अभी ये एग्जांपल हमने देखा ना, in the recombinant viral vector vaccine we use DNA of the pathogen. It is right here. We used, in this case we were using the pathogen's DNA, right? So this is a DNA vaccine delivered through a recombinant viral vector. DNA vaccine delivered through a recombinant viral vector. So this could be DNA vaccine or it could be RNA vaccine. RNA vaccine we will just see. It could be DNA vaccine or RNA vaccine. This is using the genetic material of the pathogen. Which genetic material are we going to use? That genetic material that is coding for the antigen protein. Using the genetic material of pathogen to generate vaccine. Now mRNA vaccine. Okay. mRNA vaccine is a type of vaccine that uses a copy of messenger RNA. We are using the mRNA of the pathogen to generate antigen. Now let me show you how this is happening. mRNA vaccines became very famous in COVID because many COVID vaccines like Pfizer vaccine, Moderna vaccine were mRNA based vaccines. So let's see how a mRNA vaccine works. This is the coronavirus. Why is it called a coronavirus? Because its spike proteins look like a crown. What are spike proteins? Let's see. This is a coronavirus molecule, the virus. ठीक है, वायरस के पास क्या क्या चीजें हो सकती हैं? डीएनए हो सकता है, आरएनए हो सकता है, प्रोटीन हो सकता है. Does it have enzymes for this process? Yes. Does it have own cellular machinery? No. Will it have mitochondria, nucleus? No. Will it have ability to generate energy? No. But will it have the ability to replicate? Yes. Artificially? No. Replicate where? Inside the body. So the coronavirus, what it has certain proteins here. Okay. These proteins are called spike proteins or S proteins. When COVID infects you, it is only this spike protein that the B cell sees. The B cell will look at the spike protein, say, bahar ka banda hai, maro. Foreign person, kill it. Okay. Spike proteins are therefore what? Spike proteins are the antigen in the case of COVID-19. वायरस है, वायरस में कुछ स्पाइक प्रोटीन्स, स्पाइक प्रोटीन्स को बॉडी के सेल्स देख के कहते हैं कि ये दिस इज अ फॉरेन मॉलिक्यूल, आई नीड टू किल इट. सो स्पाइक प्रोटीन्स इन कोविड आर द एंटीजन. व्हाट इज द प्रिंसिपल ऑफ वैक्सीन? डिलीवर द एंटीजन, रिमूव द पैथोजन. सिंपल प्रिंसिपल. डिलीवर द एंटीजन, remove the pathogen. I have to deliver the antigen, remove the pathogen. Clear? I cannot afford to give the COVID molecule because if I give the COVID molecule, you will get COVID. So deliver the antigen, remove the pathogen. What are we doing? This is a coronavirus. So I looked inside this coronavirus. I looked inside the coronavirus. Coronavirus had the same thing. DNA to mRNA to protein. First process with C, transcription. Second process, translation. Coronavirus was doing this. Now this spike protein is a protein. It is a protein. So there must have been certain mRNA. there must have been certain mRNA that was coding for this protein. So I looked at the virus. And I found that mRNA in it. I found that particular mRNA, that sequence of mRNA that was coding for the spike protein. Now what did I do? Once I found this mRNA sequence, I used a coating of lipids. Lipid means fat. I used a coating of lipid around that mRNA sequence and delivered that mRNA sequence coated with lipid as a vaccine to this person. This person got the mRNA sequence inside his cells. Inside his cells, the mRNA sequence was there. Now, what the mRNA sequence does? Ribosomal RNA and transfer RNA are there. Ribosome will see this mRNA and say, okay, this is an mRNA. I don't know whether it is my mRNA or somebody else's mRNA, because RNA will have the same 4 molecules every place, A, U, G and C, whether it is human, rice, virus, whatever. So it saw some sequence A, U, G, C, etc. So the ribosome saw this mRNA and started making these spike protein. Now my own cell has spike proteins. So my B cell says, okay, this is a foreign cell, let me kill it. So then the B cell started generating antibodies against the antigen of spike protein. Then my B cell generated memory. My B cell had memory. So later when the actual coronavirus came, I was protected. This is how mRNA COVID vaccine works. Okay. Now what I can figure out, you must have 2 questions. One, why coating of lipid is there? Okay. And you already know the answer to it. Let me just refresh your memory. When we were reading the plasma membrane or cell membrane, right? It was looking something like this. What did I tell you? It was selectively permeable. Selectively permeable. If I put a RNA molecule on top of the plasma membrane of a cell, if this is a cell, if I put just RNA molecule here, it will not be able to enter the cell. But if I coat this RNA molecule with lipid, if I do something like this, lipid coated RNA, then it will be able to enter, because the plasma membrane or the cell membrane is selectively permeable. It is permeable to a lipid coated RNA. It is not permeable to only RNA. Okay. So this lipid coated RNA enters the cell. Now you must be asking that here there is no vector, there is no integration happening. ये तो DNA में जाके integrate हुआ ही नहीं, है ना? ये तो ऐसे directly आ गया. तो now let's look at the cell again. Normal cell में क्या होता है? न्यूक्लियस में पोर्स होते हैं, न्यूक्लियस में डीएनए होता है, यहां डीएनए बनता है एमआरएनए. डीएनए डबल स्ट्रैंड होता है, एमआरएनए इज सिंगल स्ट्रैंड. Normally after the mRNA is transcribed, after the process of transcription, it comes out into the cytoplasm. This mRNA then is acted on by the ribosome, which codes for the protein. What am I doing in the case of this vaccine? I am taking an mRNA coated with lipid. I let it enter. It entered the cell like this. Now, how will the ribosome know this is my mRNA, this is foreign mRNA? Whatever mRNA it sees, it will be translating it into protein. Ribosome's job is translate the mRNA into protein. It is a mechanical thing. It does not have a brain of its own. It can only see A, U, G, C. So it sees AUGC here. It says, okay, I will make the protein. It sees AUGC here. It says, okay, I will make the protein. So here, what protein are we forming? Spike protein. We formed the spike protein, and spike protein was then made. Now this cell appears with spike proteins. So my B cell will say, this is a foreign cell. I must generate antibodies and I must generate memory. So it generated memory. Now when actual COVID infection happens, I will have memory. So this is how the mRNA vaccine for COVID works. Okay. I hope this is clear. If it is not clear, just read this. This is written very, very lucidly, but I cannot make it more clear than this. Okay. So now there is a second or new type of self-amplifying mRNA vaccine. This you will see in this year's current affairs. But the problem is the current affairs will not explain to you what an mRNA vaccine is. So you don't know what an mRNA vaccine is. How will you know what a self-amplifying mRNA vaccine is? In mRNA vaccine, what was I doing? I was giving small mRNA coated with lipid into the cell that forms the antigen. Okay. In self-amplifying mRNA, along with that part of the mRNA that codes for protein, I am also giving some other mRNA that codes for some other proteins. So in normal mRNA vaccine, what I was doing was, I was giving only the mRNA sequence. I was giving only the mRNA sequence that codes for the antigen. This mRNA sequence was coated with a lipid. Okay. In self-amplifying mRNA, I am giving that mRNA sequence, but I am also giving 4 other proteins. Some proteins and some mRNA. Why am I giving these proteins? Because this mRNA that I am now made, which has information for a particular antigen and information for some other proteins as well, what will it do? It has information for antigen A, and information for some other proteins. 4 other proteins, their names are not important. So when my ribosome starts translating, first it will translate this and say, okay, protein A is here, this is the antigen. Then it starts translating this protein 1, protein 2, protein 3, protein 4. What is the job of these 4 proteins? They are enzymes that help in the replication of this. Okay, how? Let's see. I give this self-amplifying mRNA I have given to the cell. It engages with the ribosome. Ribosome translates the target protein. Target protein is the antigen. It translates the target protein, antigen is made. It also translates 4 other proteins. What do these 4 other proteins do? These 4 other proteins go and attach to this mRNA. These 4 other proteins, these 4 other enzymes get attached to the original mRNA and they start making copies of this mRNA. Now what will happen? Again my ribosome will now have 4 mRNA. So it will code for 4 antigens and 16 such proteins. Then it will have 16 mRNA and 64 such proteins, right? So this replication will happen after over and over because I am giving the information for the replication protein also with the mRNA. Now why am I doing this? I don't want or I don't have the ability to get a lot of mRNA out. Even I don't have the ability to get 0.5 ml of mRNA out. So in this case, I can give very less mRNA and rest it will replicate by itself. तो बहुत कम mRNA देके भी by giving very little mRNA, I will be able to make lots and lots of protein. Which protein? Antigenic protein. Because one mRNA molecule by itself will make many mRNA molecules. So many antigenic proteins will be formed. This is a new type of technology called self-amplifying. Word breakdown करते हैं. Self-amplifying, जो खुद को बढ़ाता रहे, जो खुद को ही कॉपी करके फीचर बनाता रहे. Self-amplifying, copying itself, increasing its own number. Self-amplifying mRNA. This is a new technology. This is in this year's current affairs. So you know what mRNA vaccine is. You know what self-amplifying mRNA vaccine is. Okay. All right. Now let's see DNA vaccine. As soon as we finish vaccine, I'll give you a break. Now let's see DNA vaccine. Again, same thing we are doing. DNA encoding the antigen is inserted into the bacterial plasmid. We had seen RNA vaccine. We have seen DNA vaccine also. This is exactly what a DNA vaccine is. What I have drawn for you here. Okay. Take the DNA out that codes for antigen, get it into a vector and then let the vector go to the cell, let the cell then manufacture that antigen, and then let the immunity take place. Okay, this is what DNA vaccine is. Because I am using DNA to deliver the antigen, remove the pathogen. Same vaccine principle, only antigen must go, so I am using the own cell's machinery to make this antigen. Okay, now the vector that I can use is either some virus, or the other vector we have read is bacterial plasmid. Bacterial plasmid is also one of the vectors we have read about. So then what we are doing, we are taking the DNA plasmid, we insert it into the muscle. Okay. Now I have told you that the plasma membrane or the cell membrane is selectively permeable. It does not let everything enter. It is selectively permeable. So if it is selectively permeable, I need something or some process to let this DNA plasmid inside the muscle cell. That process is called electroporation. What is electroporation is not important. It is only a process to let this DNA plasmid enter the cell. Now plasmid has entered the cell. What is going to happen? DNA to RNA, RNA to protein. Which DNA is there? DNA that codes for, DNA that has the information for the antigen. So my cell is going to produce the antigen. Okay, let us see a video on DNA vaccines.
So, how do DNA vaccines work? This type of vaccine takes the part of the virus RNA that is responsible for reproducing the spike proteins. Then, it makes it more stable by attaching it to some DNA from a simple virus that doesn't affect humans. This DNA section is then transferred to a plasmid. This plasmid is then injected into us and it enters the human cell. It then makes its way to the cell nucleus and enters it. Once inside, the plasmid produces mRNA, which reproduces the only thing it can, spike proteins. When the cell dies naturally, the spike proteins are found by the immune system and it begins to build the manufacturing process for new antibodies. So, if and when a real COVID-19 virus is found, our immune system can immediately produce loads of antibodies and kill it before it reproduces.
So I hope it is clear. It was fairly simple. What we had read, वही बताया था इसमें, है ना? So this is DNA vaccine. DNA vaccine, taking the DNA, encoding the antigen in the bacterial plasmid. All right. Now we have to see COVID-19 vaccines, how they are made. First COVID-19 vaccine is viral vector vaccine. In this viral vector vaccine, what have I done? बहुत सिंपल है, अब आप पढ़ चुके हो. Just, and this slide is directly from the WHO website. Okay. In what am I doing in viral vector vaccine? I am taking the genetic material that codes for the spike protein. I am integrating it into a harmless virus. How have I made the virus harmless? By cutting the part of virus that causes disease. Then this viral vector is ready. This is what the viral vector will look like. This is what the viral vector will look like. Viral vector is ready now with spike protein. Okay. Viral vector vaccine then infected into human cell. Human cell will start making the spike protein, antibodies against spike protein. First type of COVID-19 vaccine is viral vector vaccine. I am taking the genetic material, putting inside harmless virus, getting the harmless virus inside the cell, letting the cell make the spike protein, and then have antibodies. Simple. Viral vector vaccine. Second type of COVID-19 vaccine is mRNA vaccine. What am I doing here? I am taking the mRNA that codes for the spike protein. Then I am taking this mRNA, I am infecting the human cell. I am coating this mRNA with a lipid and letting it come inside the human cell. What will happen? The human cell will start making spike protein, antibodies will hit, and immunity is developed. Second type of COVID-19 vaccine is mRNA vaccine. I have taken the mRNA, I have put it inside the vaccine after coating it with lipid, then letting it come to the cell, allowing the cell to make the COVID-19 spike protein, then spike protein will be infected with antibodies and the body will be able to generate memory and immunity. Third type. Third type is the subunit vaccine. In this case, I am able to identify that, okay, this is the spike protein that is needed. So, I am still unable to get the spike protein directly. वो ability नहीं है अभी हमारे पास कि हम spike protein को directly निकाल लें, right? But what will I do? I will use bacteria to culture the spike protein. अब ये बात समझो क्या हो रहा है यहाँ पे. Earlier also we had seen that we were able to make insulin inside bacteria. Here I will take that part of DNA or RNA that codes for spike protein, put it inside bacterial plasmid, then put the plasmid back in bacteria. जो हम इंसुलिन के लिए कर रहे थे, है ना, हमने वापस बैक्टीरिया में डाला. Now bacteria will produce lots of spike protein. अब using the process of downstream processing, like we had read, मैं बैक्टीरिया में से spike protein निकाल लूं. Right? Then I give this spike protein as it is to the human. Now spike protein will be given to the human, immunity will be generated. Ultimately, point is I have to get the spike protein to the human. Either I can take that part of DNA that codes for the spike protein, put it in a viral vector, give it. I can take the mRNA that codes for spike protein, coat it with lipid and give it. Or I can find the spike protein itself and give it. First case is a viral vector vaccine. Second case is a mRNA vaccine. Third case is a subunit vaccine. There is also one more way. If possible, I can kill the COVID-19. If possible, I can kill the COVID-19 and it will have spike proteins. So what will this be? This will be inactivated vaccine, like we had read earlier. I kill the microbe is still there, but it has spike protein, so immunity is generated, right? So these are the methods to make COVID vaccines. Again, I'm repeating, you can put it in a viral vector, the DNA that codes for the spike protein. Ultimate aim is to get the spike protein to the cell, to the blood. So firstly, I can use a viral vector. Secondly, I can use an mRNA coated with lipid. Thirdly, I can use the subunit itself. Fourthly, I can kill the COVID and send it. So 4 different types of COVID-19 vaccines. Now we have read Covishield. Covishield की क्या technology है? What is the Covishield technology that is made by Oxford and AstraZeneca? Okay, Oxford AstraZeneca vaccine in collaboration with Serum Institute of India. What is the technology here? Viral vector vaccine. Covishield is viral vector. It puts the DNA for coding of spike protein inside a harmless virus and lets your body get it. So Covishield is what? Viral vector vaccine. Okay. Second, Covaxin. Covaxin made by Bharat Biotech. This has inactivated virus technology, meaning virus is killed, spike protein is there. Covishield was viral vector. Covaxin is inactivated virus technology. Then you must have heard about Sputnik 5, the Russian import. Sputnik 5 is also similar to Covishield. Okay, same viral vector technology. Then we have 2 other newly developed vaccines which you will find in this year's current affairs, ZyCoV-D and Inocovac. Let's look at them. ZyCoV-D, this was developed by Zydus Cadila. It is an Indian company. It is the world's first DNA vaccine against COVID-19 using bacterial plasmid. Okay, DNA vaccine against COVID-19, the first ever is ZyCoV-D. Uses a bacterial plasmid. ZyCoV-D DNA vaccine. This has been developed in partnership with Department of Biotechnology under Mission COVID Suraksha. Mission COVID Suraksha in partnership with Department of Biotechnology, a company called Zydus Cadila has made the world's first DNA vaccine using a bacterial plasmid against COVID-19. This can be given to adults It can be given to those who are even 12 years old. This is the advantage. It needs 3 doses. Okay, 3 doses are needed to generate the proper immunity. It has a new system called TROPIS. TROPIS is a system that allows to give the vaccine without a needle. What is there? Basically इसमें क्या है कि इसमें एक jet system है. It is a very high powered jet that percolates your skin. Skin के just नीचे जाता है और फिर आपका काम चल जाता है. The cell is delivered. So exactly जो traditional needle होती है वो नहीं लगती. It is a TROPIS jet based system to penetrate the skin. कैसे काम करता है is not important for us. We just need to remember that there is a TROPIS jet based system in ZyCoV-D. D for DNA. It is the world's first DNA vaccine against COVID-19. Okay, ये हो गया. Now last vaccine is Incovac. Incovac has also been developed by Bharat Biotech, the ones that have developed the Covaxin. They have developed the Incovac. It is the world's first intranasal vaccine. Intranasal vaccine means if you just have to take it through your nose, it does not need needle injection. You take it through your nose and then it will go to the blood and the background process will happen. Okay, so this is the world's first intranasal through the nose vaccine for COVID-19. This has also gotten emergency use authorization and it is, ये तो administration हुई. Intranasal is the administration. You can see in the name here, IN, intranasal. But it is based on same viral vector technology. Okay, this is Incovac. So this ends COVID vaccines for us. There are some other vaccines also that we have to study. Next we have pneumococcal vaccine. Now this is not a COVID related vaccine, okay? This is against this. Pneumococcal vaccine. Pneumococcal vaccine is against a bacteria called Streptococcus pneumoniae. It's not important कि आप इसका नाम याद रखो, but इस वैक्सीन के बारे में क्या है वो आपको याद करना है. It is against a bacteria called Streptococcus pneumoniae. All right. It causes, this bacteria Streptococcal pneumoniae causes pneumonia, causes meningitis, many other diseases हम देखते हैं. Why is pneumococcal vaccine in the news? Pneumococcal vaccine is in the news. Firstly, it is a conjugate vaccine. What is a conjugate vaccine? It combines weak antigen and strong antigen. ठीक है? It has been included in the universal immunization program. The universal immunization program is the flagship program of the government for immunization. It covers 12 vaccine preventable diseases. Universal immunization program covers 12 vaccine preventable diseases. उसमें हमने pneumococcal conjugate vaccine को भी include कर दिया. What are the diseases that are covered by the universal immunization program? These diseases are DPT, standing for diphtheria, pertussis, tetanus. We will understand what this means, what these diseases are tomorrow. It has polio. ये तो आपने सब लिया ही होगा, सबने ये वैक्सीन. It has MMR, measles, mumps and rubella. DPT, MMR, polio. Then it has a vaccine called BCG. BCG vaccine prevents against tuberculosis of a severe form in children. Okay. Then it has a vaccine called rotavirus. Rotavirus vaccine is against this rotavirus that causes diarrhea. Okay. Then it has hepatitis B. Hepatitis B is a disease of the liver. ये diseases हम कल पढ़ेंगे. And lastly, it has HIV. HIV stands for Haemophilus influenza type B. Haemophilus influenza type B. So how many diseases are being covered? Diphtheria, pertussis, tetanus 3, polio 4, MMR, measles, mumps, rubella 7, hepatitis B, hib, rotavirus, BCG. This is 11. Okay. Now mumps, this second disease, this is no longer covered because of its less prevalence. तो कितनी हो गई? 10. 10 diseases are covered in universal immunization program. Diphtheria, pertussis, tetanus, polio, measles, mumps not included, rubella, hepatitis B, hemophilus influenza type B, rotavirus and TB through BCG. Hemophilus influenza type B क्या करता है? Hemophilus influenza type B can cause two diseases, either pneumonia happens in the lungs, or meningitis. Meningitis happens in the brain. All of this information is written in the slide. So these are 10 diseases covered in India's universal immunization program. But यहाँ तो 12 लिखा है. So the two other diseases that are covered are not included with these 10. They are counted differently. Why? Because ये 10 की 10 nationally cover होती हैं. Every child will get all of these 10 vaccines to prevent against these 10 diseases. Okay. So there are 10 nationally covered vaccine preventable diseases and 2 sub-nationally covered vaccine preventable diseases. Sub-national means ये सिर्फ वहां दिया जाएगा जहां इसकी prevalence ज्यादा है. Sub-national diseases जो covered हैं इसमें 2, उसमें पहली है pneumococcus वाली disease, ये PCV, pneumococcal conjugate vaccine, and the second is the disease of Japanese encephalitis, जो आपने सुना होगा जिससे बिहार में कई बार बच्चों की डेथ होती है, राइट? सो दिस जापानीज एंसेफेलाइटिस डिजीज इज कवर्ड एंड pneumococcal disease is covered sub-nationally. It is not covered throughout India. Sub-nationally, ये जो pneumococcus है, this is administered in Bihar and Himachal Pradesh, and Japanese encephalitis is administered in those areas where it is seen कि बहुत ज़्यादा prevalence है. Okay. Now, pneumococcal conjugate vaccine has been included in UIP. This is why it was in news. UIP हमने cover कर लिया, Universal Immunization Program. Now there is another program called, ये आप प्लीज लिख लो, मिशन इंद्रधनुष. सुना भी होगा ये आपने, इंद्रधनुष. इंद्रधनुष इज हिंदी फॉर रेनबो. तो इंद्रधनुष कवर्स 7 वैक्सीन प्रिवेंटेबल डिजीजेस. इसको हम डिटेल में कल देखेंगे, बट मिशन इंद्रधनुष कवर्स 7 वैक्सीन प्रिवेंटेबल डिजीजेस. ये बेसिकली इसके लिए बनाया गया था कि जो बच्चे इस यूनिवर्सल इम्यूनाइजेशन प्रोग्राम से छूट गए हैं, हम उनको भी वैक्सीन दे दें. राइट, कैचअप प्रोग्राम था ये मिशन इंद्रधनुष. ओके. नाउ न्यूमोकोकल कॉन्जुगेट वैक्सीन पीसीवी, इट प्रोवाइड्स प्रोटेक्शन अगेंस्ट स्ट्रेप्टोकोकस निमोनिया व्हिच कॉजेज व्हाट. यहां पे लिखा है इट कॉजेज निमोनिया, इट कॉजेज मेनिंजाइटिस, साइनसाइटिस, ब्रोंकाइटिस एंड मिडिल ईयर इन्फेक्शन. डोंट वरी, सब बताता हूं. निमोनिया, वो तो नाम से ही पता चल रहा है, न्यूमोकोकल वैक्सीन. निमोनिया, निमोनिया मींस लंग इन्फेक्शन. दैट्स ऑल यू नीड टू नो. लंग्स में इन्फेक्शन इज निमोनिया. मेनिंजाइटिस, मेनिंजाइटिस हैपेंस इन द ब्रेन. जो हमारा ब्रेन होता है, लुक समथिंग लाइक दिस, है ना, इसके बाहर जो एक कवरिंग होती है, इसे हम कहते हैं मेनिंज. So there are some coverings in the brain called meninges. When these coverings get inflamed, when they get infected, it is called meningitis. So streptococcus pneumonia can A cause pneumonia, 2 it can cause meningitis, C it can cause sinusitis. जब हम breathe करते हैं, if you touch here and here, your body has certain hollow areas inside the bone called the sinus. वहां इन्फेक्शन हो जाए तो उसे कहेंगे साइनसाइटिस. ये भी करा सकता है स्ट्रेप्टोकोकस निमोनिया. फिर है ब्रोंकाइटिस. व्हाट इज ब्रोंकाइटिस? व्हेन वी आर ब्रीदिंग, आवर लंग्स हैव दिस स्ट्रक्चर. ऐसे दिखते हैं हमारे लंग. यहां से हमारा नोज से एयर आती है. दिस इज द ट्रैकिया, द बिग फूड विंड पाइप. विंड पाइप इज ट्रैकिया. देन इट गेट्स स्प्लिट इनटू 2 पार्ट्स. दिस इज वन लंग, दिस इज द अदर लंग. तो ये जो है, जो लंग के अंदर जाती है, इसे हम कहते हैं ब्रोंकस. तो वो या तो वो निमोनिया करा दे, यानी वो लंग को इन्फेक्ट करा दे तो होगा निमोनिया. अगर वो इस ट्यूब को इन्फेक्ट करा दे तो होगा ब्रोंकाइटिस. तो स्ट्रेप्टोकोकस निमोनिया कैन ईदर इन्फेक्ट द लंग causing pneumonia, it can infect the bronchus, it can infect your meninges causing meningitis, okay, and it can also cause middle ear infection. middle ear means आपका ईयर में 3 पार्ट होते हैं, आउटर ईयर, मिडल ईयर, इनर ईयर. तो जहां हियरिंग होती है दैट इज द इनर ईयर, जहां ईयर ड्रम के बाद वाला पार्ट होता है दैट इज द मिडल ईयर. इतना आपको डिटेल जानने की जरूरत नहीं है, बट मिडल ईयर इन्फेक्शन. तो हमने 4 चीजें देखी जो स्ट्रेप्टोकोकस निमोनिया करा सकता है: निमोनिया, मेनिंजाइटिस, साइनसाइटिस. साइनसाइटिस वी हैव सीन, साइनसेस यहां होते हैं, ब्रोंकाइटिस एंड मिडल ईयर इन्फेक्शन. Pneumococcal vaccine are of 2 types. First is pneumo vaccine, which are only polysaccharide. Polysaccharide means only have carbohydrates. ऐसे. बट इसमें क्या है प्रॉब्लम? इसमें इम्यूनिटी स्ट्रांग नहीं आएगी, इम्यूनोजेनिसिटी कम होगी. तो पॉलीसैकेराइड वैक्सीन हमें बनाना नहीं है. वाई? बिकॉज इट डज नॉट हैव इनफ इम्यूनिटी जनरेटिंग एबिलिटी. Because polysaccharides, like we read, are not proteins. Polysaccharides are carbohydrates. So polysaccharides are weak antigen. So what we do, if we make a polysaccharide vaccine, जो बच्चे हैं, बच्चों की तो इम्यून सिस्टम वैसे ही वीक है, दे डोंट हैव दैट इम्यून जनरेटिंग एबिलिटी. हमने दे भी वीक एंटीजन दिया. अब ये मत समझना कि वीक ही देना है बच्चों को. दिस इज नॉट पैथोजन, दिस इज एंटीजन. इट डज नॉट कॉज डिजीज, right? But we have to give a strong antigen. Strong antigen meaning which has the ability to generate lots of antibodies. तभी तो बच्चा immunity develop कर पाएगा. Only then the child will be able to get immunity. So what we do, we give a conjugate vaccine where we combine a protein antigen with a carbohydrate antigen. We combine these two. This is a pneumococcal conjugate vaccine. Okay, this is why it's called PCV, pneumococcal conjugate vaccine. If only polysaccharides are present, the immunogenicity is less. So carrier protein is attached, polysaccharide plus protein forming conjugate. Okay, so that immunogenicity is strong. अब ये होती डिजीज बच्चों में है तो हमें बच्चों को ही तो देना है. If we give only polysaccharide, कोई पर्पस नहीं रहेगा. That is why it's a conjugate vaccine. Now we are seeing BCG. BCG क्यों न्यूज में है? Because 100 years हो गए हैं इस वैक्सीन को बने हुए. BCG is Bacillus calmette-guerin. BCG vaccine is to protect against a childhood severe TB. Severe tuberculosis in childhood से प्रोटेक्ट करता है बीसीजी वैक्सीन. टीबी का पॉजिटिव ऑर्गेनिज्म जो है उसका नाम होता है माइकोबैक्टीरियम ट्यूबरकुलोसिस. इट्स रिटन हियर, माइकोबैक्टीरियम ट्यूबरकुलोसिस इज द पॉजिटिव ऑर्गेनिज्म इन बीसीजी. ठीक है, इन टीबी. माइकोबैक्टीरियम ट्यूबरकुलोसिस कॉजेज द डिजीज ट्यूबरकुलोसिस. दैट इज व्हाई द डिजीज इज कॉल्ड ट्यूबरकुलोसिस. इसके अगेंस्ट जो वैक्सीन है उसका नाम है बीसीजी. BCG doesn't protect everybody from TB. It just protects children from severe TB. So it's possible that you get the BCG vaccine like we have all gotten and still get TB. But in childhood you will be protected from severe tuberculosis. अब इसमें एक बहुत ही स्पेशल बात है कि the farther you go from the equator, this is the earth, this is the equator, this is India somewhere, this is the Tropic of Cancer, right? The farther away you go from the equator, जितना दूर आप जाओगे, यहां पहुंच जाओ somewhere around Finland, या यहां पहुंच जाओ around Canada, right? या यहां around Russia. The farther you go from the equator, the better the BCG vaccine will work. In India, its working will be less. वहां it will be more. Why? India में क्या है? Because of the environmental conditions, there are many strains of Mycobacterium tuberculosis. तो सारे strains के against it is not effective. What strains are, we will see tomorrow when we do viruses. It is not effective against all the strains in India. But वहां क्या है कि strain ही एक है, so the effectiveness is much more. Due to the environmental presence of different strains in tropical countries, BCG's effectiveness is less. But BCG's effectiveness is very high in these areas. तो अब आप सोच सकते हो कि ये जो कंट्रीज हैं, रिच कंट्रीज सारी इधर ही हैं वर्ल्ड की, उनमें बीसीजी काम करता था. इंडिया में और अदर ट्रॉपिकल कंट्रीज में, अफ्रीका में, इट यूज्ड टू वर्क बट नॉट दैट गुड. तो आज तक बीसीजी के अलावा कोई वैक्सीन ही नहीं बना जो टीबी से प्रोटेक्ट कर सके. बट जो डिजीजेस वहां होती हैं, उन डिजीजेस के लिए वैक्सीन बन जाता है. कोविड के लिए वैक्सीन 1 साल में बन गया. टीबी के लिए वैक्सीन इतने सालों से नहीं बना, because TB was not infecting these people, and they are the people with all the money. So there is an idea you will see in the PT 365 also called neglected tropical diseases. This is why neglecting of tropical diseases happens, because all the money in the world is concentrated in these latitudes. So in IR we use a term called the global north. Even Australia is part of the global north. Even Japan is part of the global north. Even New Zealand is part of the global north. Why? Because these are those countries that are rich, developed, industrialized, and they do not care about the interests of countries here which are developing or least developed. So the countries in the global north do not get infected with TB. So there is no vaccine that protects completely from TB, but all of them were facing COVID, so they made vaccines, and so many vaccines. मैं पढ़ाते पढ़ाते थक गया. So many types of vaccines against COVID. Why? Because all the money in the world is here, because these were the countries that practiced colonialism mostly, right? They have earned the money and they control how scientific research goes. That is why in our G20 presidency there is a priority called Voice of the Global South. There is a global south, countries of Africa, countries of Asia, countries of Latin America that are still developing, and their concerns are not heard in the world. So India is trying to become a leader of these countries. This is called Voice of Global South. The problem is global north, since they invest only in their own interests. Therefore, there is an idea called neglected tropical diseases. Okay, this term you will see in PT 365, but the explanation is not there. This I am giving you right now. Okay, BCG's effectiveness is farther from the equator because tropical regions have environmental strains of Mycobacterium. Now, BCG is against Mycobacterium tuberculosis. Okay, Mycobacterium tuberculosis. The bacteria that causes leprosy is called Mycobacterium leprae. So since they are similar, chickenpox, smallpox, since they are similar, Mycobacterium leprae causes the disease leprosy. So even against Mycobacterium leprae, the BCG vaccine works. So the disease like leprosy can also be dealt with BCG vaccine. It is effective against this also. It is not the vaccine of choice, but it has certain effectiveness. Then there is a disease called Buruli ulcer. You don't need to know what it is. ये आपको मगअप करना होगा कि BCG is used against Buruli ulcer, and in case of bladder cancer also, we can use BCG. How? I don't want to explain here. It will take an hour. Okay. So BCG can be used against tuberculosis, leprosy, Buruli ulcer, and bladder cancer. India in the world has 27% of the global disease burden of tuberculosis. We do not have 27% of the world's population, but we have 27% of the world's tuberculosis cases. That is why in India there is a constant push to eliminate this disease. And recently in the budget there was announced, and it has been in the works for some time, called the Prime Minister's TB Mukt Bharat initiative. Okay, TB हम कल पढ़ेंगे. It's in the news also, but I'm just giving you a background. Now, India has also earned certain research money. We are developing, our country is growing. So the Indian Council of Medical Research is now taking trials of recombinant BCG vaccine. Recombinant BCG vaccine, which is called VPM 1002. Name is written here also in the one pager also. And MIP vaccine called Mycobacterium indicus prani. Again, you don't need to know the detail here, just the name. VPM 1002 and MIP, these are new vaccines that the Indian Council of Medical Research, ICMR, is trialing. Okay. Now there is toxoid vaccine. Toxoid vaccine. What is toxoid? First, we have to understand toxoid. Only then we can understand toxoid vaccine. So toxoid is an inactivated toxin. We have also seen protoxin. So protoxin is previous stage of toxin. Before forming the toxin, what forms is protoxin. Once the toxin is formed and I inactivate it, I don't let it do its actual function, it will be called toxoid. Okay. If I suppress the toxicity, if I suppress the ability of the toxin to cause any disease or toxicity, it will be called toxoid. It can be done either through a chemical method or a heat method. या तो मैं chemical लगा दूं इसके ऊपर या मैं heat लगा दूं कि toxoid, right, the thing that causes toxicity वो खत्म हो जाए, it gets inactivated, it will be called toxoid vaccine. Toxins are secreted by bacteria, right? Toxoid is not secreted by bacteria. Toxoid I have made artificially. Toxoid is used in vaccination. Okay. Now, why is it used in vaccination? कुछ diseases में क्या होता है that the toxin itself is the antigen. What happens in some cases is that the toxin itself, the toxin that the bacteria produces inside our body, it is itself the antigen. So if the toxin itself is the antigen, I have to give the toxin, but I cannot give the toxin because it can cause damage. So I have to inactivate the toxin. How will I inactivate the toxin? I will make it into a toxoid. So once I give toxoid, it will be toxoid vaccine, inactivated toxin vaccine. Iska example is diphtheria toxoid vaccine and even tetanus toxoid, TT, is also that. Okay, TT and diphtheria toxoid vaccine. ठीक है. Now, tetanus vaccine. मुझे पता था अब आपका नेक्स्ट क्वेश्चन यही है. This is why I put it in the slide here. कि मैंने पहले तो कहा था कि ये तो पैसिव वैक्सीन होता है. अब मैं कह रहा हूं कि टॉक्सॉइड वैक्सीन है. तो टेटनस वैक्सीन इज ऑफ टू टाइप्स. वन इज एक्टिव टेटनस वैक्सीनेशन. What will be active vaccination? I will let my own body form the antibodies. This is given to children, to growing children around 10 years and 16 years it is given. It is also given to pregnant mothers. Isme what happens is in childhood it is given that the body starts forming its own antibodies. But the problem with active tetanus vaccination is that the memory does not stay for very long time. हमारे पास जो इम्यूनोजेनिसिटी है, इसकी इम्यून जनरेटिंग एबिलिटी है इस वैक्सीन की, वो काफी कम है. तो क्या होता है कि अगर आपको अभी अभी रिसेंटली टीटी लगाए, टेटनस टॉक्सॉइड लगाए एंड आपको चोट लग जाए, द बैक्टीरिया दैट कॉजेस टेटनस इज कॉल्ड क्लोस्ट्रीडियम टेटनी. दिस एंटर्स द बॉडी थ्रू स्किन ब्रेक्स. इफ योर स्किन विल ब्रेक, क्लोस्ट्रीडियम टेटनी कैन एंटर एंड इट इज यूजुअली फाउंड ऑन रस्टेड आयरन. तो रस्टेड आयरन पे अगर आप गिर गए और चोट लग गई, या सीमेंट पे गिर गए चोट लग गई, तो क्लोस्ट्रीडियम टेटनी कैन एंटर योर बॉडी. अब अगर रिसेंटली आपको एक्टिव टेटनस वैक्सीन लगाए तो कोई बात नहीं, बट अगर काफी टाइम हो गया तो अब क्या है, मेमोरी जा चुकी है. नाउ क्लोस्ट्रीडियम टेटनी इज इनसाइड योर बॉडी. आपको चोट लग गई, आप गिर गए, सो यू आर एट हाई रिस्क. नाउ क्लोस्ट्रीडियम टेटनी इज इनसाइड द बॉडी, यू आर एट हाई रिस्क. I cannot now wait for the body to generate immunity again. कि मैं दोबारा बॉडी को बोलूं कि यार मेमोरी सेल्स बनाओ, इम्यूनिटी जनरेट करो. I can't wait for that. So what will I do? I will give passive vaccination. What is passive vaccination? Passive vaccination is I give the antibodies directly. आई गिव द एंटीबॉडीज डायरेक्टली, मैं एंटीबॉडीज ही दे दूं, मैं वेट ना करूं कि आप अपनी जनरेट करोगे इम्यूनिटी वगैरह. तो इन अ हाई रिस्क सिचुएशन आई कैन गिव पैसिव टेटनस. In normal situation, I give active tetanus or TT. So these are two types of tetanus vaccine. Other example of toxoid vaccine is diphtheria. Okay. Now this is a test of 4 questions and it is not a learning exercise today. Today I expect that you will get all the questions right. ठीक है, 4 ही क्वेश्चन है. They are simple. I will expect that you get them right. Okay. So I will give you time of 1 minute for each question and then we will solve these questions. Okay. Chalo, let's go. Solve this. I know this is tough. Think and solve. Okay. Next question. Okay. So test is done. Again, firstly, congratulations. आपने टेस्ट दिया, आप बैठे. Good job. अब आंसर देखते हैं. First is, what is the importance of using pneumococcal conjugate vaccines in India? ठीक है, अब easy way to do this is you see the third statement and you say cannot be C or D, right? Now you don't need to read the first statement. Okay, now it's all about the second statement. जान के I have put this question first because this was the toughest question. बाकी तो आपको मैं बता ही चुका हूं, this is the tough question. बट आप देखो, आप लॉजिक लगाओ, डिपेंडेंस ऑन एंटीबायोटिक्स दैट आर नॉट इफेक्टिव अगेंस्ट ड्रग रेजिस्टेंट बैक्टीरिया कैन बी रिड्यूस्ड. डिड आई नॉट टीच यू अबाउट ड्रग रेजिस्टेंस, एंटी माइक्रोबियल रेजिस्टेंस? व्हाट डिड आई टेल यू? कि हमने कहा बॉल वॉर्म का एग्जांपल लेके समझा था कि अगर मैं एंटीबायोटिक खाता जाऊंगा, मैं एंटीबायोटिक पे एंटीबायोटिक खाता जाऊंगा, तो एक ना एक टाइम पे ऐसे बैक्टीरिया आ जाएंगे जो नेचुरली रेजिस्टेंट होंगे. जब वो नेचुरली रेजिस्टेंट बैक्टीरिया फैलेंगे तो एंटीबायोटिक रेजिस्टेंस हो जाएगी. देयर विल बी नो वे टू प्रोटेक्ट अगेंस्ट देम, राइट? दिस वाज द आइडिया बिटवीन बॉल वॉर्म, बॉल गार्ड टेक्नोलॉजी एंड बीटी कॉटन में भी हमने ये सेम चीज पढ़ी थी. नाउ इफ आई एम यूजिंग अ वैक्सीन, तो मैं बॉडी की नेचुरल इम्यूनिटी यूज कर रहा हूं, मैं एंटीबायोटिक नहीं यूज कर रहा. तो माय एंटीबायोटिक यूज विल रिड्यूस इफ आई यूज अ वैक्सीन, करेक्ट? If I use a vaccine, my body's natural immunity will be there. I will not need to eat medicine to kill the bacteria in my body. So if I will not need to use antibiotics, the dependence or the use of antibiotics will go down. When the use of antibiotics will go down, the antibiotic resistance will be reduced. The dependence on antibiotics will be reduced. So the second statement is correct. Dependence on antibiotics that are not effective against drug resistant bacteria can be reduced. I will reduce this dependence because I am using the body's own immune system. Answer here is B. पहला क्वेश्चन गलत करना अलाउड है, ये टफ था, हमने एंटीमाइक्रोबियल रेजिस्टेंस कल पढ़ना है, सो इट्स ओके. सेकंड क्वेश्चन गलत करना बिल्कुल भी अलाउड नहीं है. देखो, अब जो आप मेरे से क्वेश्चंस इतने पूछ रहे थे, जस्ट रियलाइज एंड सी, यूपीएससी भी बहुत बेसिक अंडरस्टैंडिंग चेक कर रहा है यार. दे डोंट वांट यू टू नो इन डेप्थ. तभी मैं आपको बिल्कुल डेप्थ में जाके एक एक चीज नहीं समझा रहा, बिकॉज द क्वेश्चंस आर सो बेसिक. रिकॉम्बिनेंट वेक्टर वैक्सीन्स, जेनेटिक इंजीनियरिंग इज अप्लाइड. यस, यू नो रिकॉम्बिनेंट तो है, तो टू गेट पास्ट. डीएनए और आरएनए दोनों के केस में मैं ऐसे नहीं कि उसको वहीं पे छोड़ दूं और वो चला जाए. तो आरएनए के केस में अगर एमआरएनए है तो या तो हम लिपिड कोटिंग कर रहे थे, करेक्ट, या तो हमने लिपिड कोटिंग कर रखी थी. दूसरा, डीएनए के केस में हम क्या करते हैं, हम वायरल वेक्टर यूज करते हैं. डीएनए के केस में हम प्लाज्मिड भी यूज कर सकते हैं, बिकॉज प्लाज्मिड इज अ वे टू कैरी, प्लाज्मिड इज अ वे टू कैरी द डीएनए. एंड प्लाज्मिड अगर हमने बैक्टीरिया के अंदर डाल दिया तो बैक्टीरिया तो क्रॉस कर ही रहे हैं ना हमारी सेल मेम्ब्रेन, दैट इज व्हाई दे आर इन्फेक्टिंग अस. तो प्लाज्मिड इज आल्सो अ वे. इवन इन आरएनए वी कैन यूज प्लाज्मिड, इवन इन आरएनए वी कैन यूज वायरस एज वेक्टर. ये हम कल सेट पढ़ेंगे व्हेन वी अंडरस्टैंड व्हाट वायरसेस आर. तब आपको डीएनए वैक्सीन और वायरल वेक्टर वैक्सीन का जो बेसिक डिफरेंस है वो समझ. ओके, ऑलराइट. सो इन दिस केस द आंसर इज सी, बोथ, सिंस बैक्टीरिया एंड वायरसेस बोथ कैन बी यूज्ड एज वेक्टर्स and genetic engineering is applied. Next, in context of vaccines manufactured to prevent COVID-19, consider the following statements. Serum Institute of India produced COVID-19 vaccine called Covishield using mRNA platform. You know this statement is wrong, right? Serum Institute of India का जो Covishield vaccine है, it is not mRNA vaccine, it is viral vector vaccine, right? So if you just eliminate the first statement, the answer is there. You don't even need to think about 2 and 3. पढ़ो भी मत, but हम पढ़ लेते हैं. Sputnik 5 based on vector based platform, we know this, and Covaxin is inactivated pathogen. This also we know. See, the questions in vaccines are basic. So अगर basic understanding भी है तो चलेगा. If I spend a lot of time here explaining DNA vaccine versus recombinant viral vector vaccine, we will miss out on something ना यार. इसीलिए मैं कह रहा हूँ कि एक लेवल से ज्यादा if we go in depth, we will have to leave something else. We don't have unlimited time, right? And that is why I am explaining you merely the basics, because you look at the level of questions they are asking. If you just understand the basics, it will be fine. These questions cannot be solved if you are mugging up. But if you have an understanding, then they can be solved. Okay. Last question. describes the role of B and T cells. 2022 के prelims का question है ये. These are all previous year questions, by the way. Just look at this, easy question. What is the role of B and T cells? Right? हम पढ़ रहे हैं immunogenicity and immunocompromised में सब यूनिट और conjugate वैक्सीन क्यों देना है, and this is what they're asking. तो आपको ये भी लग सकता है कि मैं ज्यादा पढ़ा रहा हूँ, but understand कि next time they can go slightly deeper. Right? तो एक conceptual understanding जरूरी है, but एक limit भी लगाना जरूरी है, and that finding that balance is genuinely difficult. It's difficult for you, it's difficult for me, but I am trying whatever I can to find the balance there. ठीक है? All right. So we're done with the 4 questions. Now let us go to the second part of today's class, concept of mutation. Mutation क्या है? Mutants. You must have seen the movie X-Men. That is mutants, right? There is some change. It is not normal. So mutation is change in the DNA sequence of an organism. Okay. Now why does this change happen? देखो, हमारा DNA looks something like this. Okay. Normally भी DNA में not everything is always perfect. कभी कभी कुछ breaks हो जाते हैं, कभी कभी कुछ repair होना होता है, right? Without any intervention also, sometimes let's say यहाँ पे थोड़ा सा एक break लग गया, यहाँ पे थोड़ा सा break लग गया, then what the cell का response क्या है? Cell का response है कि वो इसको अपने आप repair कर देता है. It has its own repair mechanisms to correct these breaks or these problems that might occur. Okay? But कभी कभी ये repair होते होते ठीक नहीं होता repair. जैसे मान लो क्या हुआ कि नॉर्मली डीएनए ऐसा था, A T T G C. दिस वाज द नॉर्मल डीएनए सीक्वेंस. तो सामने क्या होगा? T T A C G. ये नॉर्मल डीएनए था. अब गलती से इसमें क्या हुआ कि एक ब्रेक लग गया, लाइक अ कट हैपेंड हियर. नाउ द सेल हैड टू रिपेयर इट. So in the process of repair, यहां पर तो A वर नॉर्मल, सामने T वर ऑल्सो नॉर्मल, ठीक है. बट जब यहां T और T लगाना था सेल को रिपेयर में, उसने गलती से लगा दिया T और A. ओके. तो अब देखो डीएनए सीक्वेंस चेंज हो गई. नाउ द डीएनए सीक्वेंस इज चेंज्ड. समथिंग एल्स हैज हैपेंड नाउ. At least one DNA molecule has changed. Originally it was AATTGC and सामने उसका complement. यहाँ अब क्या हो गया? AATAGC. So some change happened because, and ये बहुत large scale पे होता है. I'm just showing 6 pairs here. This is like base pair. A and T is one pair. और सामने A के सामने T आना है, तो it's called base pair. तो मैं यहाँ पे सिर्फ 6 दिखा रहा हूँ, बट बहुत ज्यादा बेस पेयर्स है हमारे डीएनए में. तो इसमें क्या हो रहा है? इसमें कट लग गया. नॉर्मल रिपेयर में जो प्रोसेस होती है 99% टाइम कुछ नहीं होता, बट कभी कभी ऐसे गलतियां हो जाती हैं इन द रिपेयर, इन द कॉपिंग, राइट. अब देखो, अब जो हमारा डीएनए है उसमें सारा तो कोडिंग नहीं है. नॉट ऑल डीएनए कंसिस्ट ऑफ जीन. कहीं कहीं पे जीन है, राइट. अब अगर इस पार्ट के रिपेयर में मान लो यहां पे रिपेयर में कोई गलती आ भी गई, द रिपेयर मिस्टेक हैपेंड ऑल्सो, इट विल नॉट मेक सच अ डिफरेंस ना. कोई इतनी बड़ी बात थोड़ी हो जाएगी यहां पे गलती आई तो. बट इफ हेयर सम प्रॉब्लम हैपेंस, देन इट विल लीड टू अ ह्यूज इंपैक्ट, बिकॉज जो यहां से प्रोटीन बनना था वो प्रोटीन चेंज हो जाएगा. एंड इवन वन म्यूटेशन इन द प्लेस ऑफ प्रोटीन कैन कॉज ह्यूज डिजीज. वन म्यूटेशन मीन्स बस एक जगह ए था, ए की जगह टी हो गया, तो ये बहुत बड़ी म्यूटेशन हो गई. क्योंकि इससे इंपैक्ट देखो, यहां पे जीन थी, जीन में कुछ सीक्वेंस था, उस सीक्वेंस के हिसाब से क्या बनने थे? प्रोटीन. पहले एमआरएनए बनता, फिर एमआरएनए से बनते प्रोटीन. तो जब प्रोटीन बनते, व्हाट वुड बी द वे टू फॉर्म द प्रोटीन? प्रोटीन्स वुड जस्ट बी पहले एक अमीनो एसिड, फिर दूसरा अमीनो एसिड, फिर तीसरा अमीनो एसिड, बेस्ड ऑन दिस सीक्वेंस, राइट. बट यहां पे सीक्वेंस चेंज हो गई, तो पहले अमीनो एसिड लगना था 1, देन 2, देन 3. अब क्या लग रहा है? अब लग रहा है 1, देन 3. ओके. तो ये क्या हुआ? ये हो गई प्रॉब्लम. जो ओरिजिनल एंजाइम बनना था, ओरिजिनल प्रोटीन बनना था, जो रिस्पॉन्सिबल था नॉर्मल फंक्शनिंग के लिए, वो अब नहीं बनेगा. दिस इज व्हाट म्यूटेशन कैन कॉज इफ इट हैपेंस इन द जीन दैट इज इन द कोडिंग रीजन। यहां म्यूटेशन हो जाए कोई दिक्कत नहीं है, क्योंकि इससे तो कोड ही नहीं हो रहा कुछ। दिस टाइप ऑफ म्यूटेशन हैपेंस इन अ डिजीज कॉल्ड सिकल सेल एनीमिया। इसमें एक देयर इज अ जीन, चलो हम कल देखेंगे उस जीन का नाम। इन जीन देयर इज ओनली वन म्यूटेशन, एक जगह बस एक ही जगह टी बन जाता है इन सम इंडिविजुअल्स, तो उन्हें सिकल सेल एनीमिया हो जाता है। सो इवन वन स्मॉल म्यूटेशन कैन कॉज डिजीज। तो म्यूटेशन इज चेंज इन द डीएनए सीक्वेंस ऑफ एन ऑर्गेनिज्म। इट कैन रिजल्ट इन एरर्स ऑफ डीएनए रेप्लिकेशन। ये भी एक इंपॉर्टेंट चीज है। जब सेल ग्रो कर रहा है, डिवाइड करके दो बना रहा है, तो उसे अपना पूरा डीएनए रेप्लिकेट करना है, ठीक है। व्हेन द सेल इज डिवाइडिंग, इट हैज दिस डीएनए, ठीक है, सेल हैज दिस डीएनए which is organized in the form of chromosome like this. When it has to replicate, it has to first form this. Then this cell will divide into two parts. So chromosome will also divide like this. One will go to first cell, second will go to other cell. जब ये क्रोमोसोम यहां जाएगा, ये क्रोमोसोम यहां जाएगा, तो अब पूरा डीएनए एज इट इज कॉपी होके डबल होना है, तभी तो दो में डिवाइड होगा। तो इस रेप्लिकेशन में हो सकता है कोई एरर हो जाए। ओके, सो डीएनए रेप्लिकेशन में भी हो सकता है। अब कुछ केमिकल्स होते हैं, जब वो हम व्हेन वी गेट एक्सपोज टू देम, म्यूटेशन के चांसेस बढ़ जाते हैं, उनसे डीएनए कट्स और रिपेयर वाली मैकेनिज्म खराब हो जाती है। दे आर कॉल्ड म्यूटाजेंस। जेन का तो आपको मीनिंग पता ही है, जेन जनरेटिंग एंटीजन पैथोजन, म्यूटाजेन म्यूटेशन जनरेटिंग। ये कैसे हैं? इसका एग्जांपल, जब रेडियो एक्टिव बॉम्ब्स आर यूज्ड, न्यूक्लियर बॉम्ब्स आर यूज्ड, दे आर कॉल्ड म्यूटाजेंस, राइट। न्यूक्लियर बॉम्ब से रेडिएशन आती है, रेडिएशन अगर हमारी बॉडी लेती है तो म्यूटेशन के चांसेस बढ़ जाते हैं। आप कभी गूगल करना कि हिरोशिमा और नागासाकी में आज भी जो बच्चे पैदा हो रहे हैं, इवन टुडे द चिल्ड्रन बोर्न इन हिरोशिमा एंड नागासाकी, दे हैव इश्यूज इन देयर डेवलपमेंट बिकॉज दैट रेडिएशन हैज बीन पासिंग ऑन म्यूटेशंस फ्रॉम जनरेशन टू जनरेशन। ओके, एंड ऑल लिविंग सेल्स कैन म्यूटेट। ओके, viruses can also mutate. When viruses mutate, they form new strains like beta, delta, omicron. कल पढ़ेंगे। Change in DNA due to mutation will lead to new genes. Now these new genes can be beneficial also. हो सकता है मेरे बैक के जो मसल्स बनाने वाली जीन है वो म्यूटेट होके विंग्स बनाने वाली जीन बन जाए, बट दिस इज एक्सट्रीमली रेयर, इट इज नेक्स्ट टू इम्पॉसिबल। 99% म्यूटेशंस आर हार्मफुल, दे लीड टू सम कैंसर, राइट। जैसे हमने क्या पढ़ा था, ऑन्कोजीन है। इट इज पॉसिबल कि नॉर्मल जीन में एक म्यूटेशन हो जाए और फिर उसमें ऑन्कोजीन वाली सीक्वेंस आ जाए तो कैंसर जनरेटिंग हो जाए, राइट। सो म्यूटेशन ये होता है। आई होप द कंसेप्ट इज क्लियर। अब हम समझते हैं समथिंग कॉल्ड जीन थेरेपी। जीन थेरेपी समझने से पहले हमें दो तरह के सेल समझने की जरूरत है। फर्स्ट इज सोमैटिक सेल, सेकेंड इज जर्म लाइन सेल। ये इस पीपीटी में नहीं है, बट इससे पहली क्लास में जो पीपीटी है उसमें आप जब डाउनलोड करोगे आपको दिख जाएगा, बट मैं यहां समझा रहा हूं। इट इज देयर इन लास्ट क्लास पीपीटी। ओके, दिस इज मी, या चलो दिस इज मिस्टर ए। नाउ व्हेन दिस पर्सन हैज टू रिप्रोड्यूस, व्हेन दिस पर्सन ए हैज टू फॉर्म sperms, right? When this person has to form sperms, they will be formed in a specific part of the body called the testes, right? These sperms will have what? Half of the DNA. They are what? Haploid cells. है ना? ये बंदा है, इसने sperms बनाने हैं। कहाँ बनेंगे? In its testes. टेस्टिस में जो स्पर्म्स बनेंगे दे विल बी हैविंग हाफ डीएनए हैप्लॉइड। ये बात हम पढ़ चुके हैं। तो अब इसका जो ब्रेन सेल है लेट्स से, वो तो 2n ही है ना, डिप्लॉइड ही है। इसके टेस्टिस में जहां स्पर्म बन रहे हैं वहां पे हैप्लॉइड सेल बन रहे हैं, राइट। सो व्हेन ही इज रिप्रोड्यूसिंग, जो डीएनए ये आगे दे रहा है इज द डीएनए दैट इज प्रेजेंट इन द टेस्टिस सेल्स, क्योंकि वहीं से these sperms are being formed and this sperm will go on to form the new baby. तो जो सेल्स बॉडी के are not involved in the meiotic process of reproduction, the cells of the body that are not involved in the process of meiosis, जो meiosis करके gametes नहीं बना रहे, sperm या ovum नहीं बना रहे, those cells are called somatic cells. Somatic cells are those that are not doing meiosis, they are not forming gametes, they are somatic cells. The other type of cell is germ-line cells. Germ-line cells are those cells that are doing meiosis to form gametes. They are doing meiosis. They are going from 2n to n. They are forming haploid cells. So there are cells called spermatocytes that generate sperms. They are germ-line cells. They germ-line. Okay. These are ones doing meiosis leading to formation of gametes. So germ-line cells, somatic cells, these are the two types of cells in your body. The cells in my hand are somatic cells. The cells that are responsible for producing sperm are germ-line cells. Germ-line cells are responsible for reproduction. अब एक आंसर दो. The question is, a mutation happens in my neck cells. A mutation has happened in my body's neck cells. Will this mutation be passed on to my children? Yes or no? A mutation has happened in my neck. Will this cell pass this mutation on to my children or not? Tell me. भाई yes कौन बोल रहा है यार? Good. Of course not. If this mutation had happened in my testicular cells, then it would pass on, because it is the testicular cells that will produce the sperm. The sperm will produce the zygote. In the neck cells or any other somatic cell, the mutation will not pass on. Only from the germ cells, the mutation will pass on. Okay. Now let us understand what gene therapy is. If a child is born with genetic defects, okay, genetic defect meaning one of my important genes, one important gene is not functioning properly, then what do? I have to give this normal gene to the child. मुझे बच्चे को normal gene देनी पड़ेगी. Okay. Because if one of the genes is defective, he will have some issue. Genetic defect. I have to give the normal gene. How? The idea is gene therapy. Gene therapy is a, मतलब बहुत सारे methods हैं इसमें, अभी हम discuss करेंगे. It is a collection of methods that allow for the correction of a genetic defect. You diagnose that this child does not have some gene or the genes are not functioning properly. I can give a gene from outside, correct that defect. Okay. Genes are inserted into a person's cells to correct the defect. Correction of genetic defect will involve what? Delivery of normal gene to the cell. Normal gene has to be delivered to the individual to take over the function of the defective gene. This is gene therapy. How is it done? We'll see. Now, gene therapy is obviously part of biotechnology. It is use of biological technology to make my life easier. It is a biotechnological method to replace defective gene with correct gene. There are two types of gene therapy: somatic cell gene therapy and germline gene therapy. If it is germline gene therapy, it will be passing on to my children as well. If it is somatic gene therapy, it is not going to pass on to my children. This is clear. Now here are some examples of somatic gene therapy. First is hemophilia. Hemophilia loves to bleed, does not have the gene to form a particular protein responsible for clot. Last time समझाया था, दोबारा नहीं बताऊंगा. Hemophilia, you don't have a particular factor that is responsible for clot formation. So you give the gene for clot formation, so hemophilia will be cured. Sickle cell anemia. Sickle cell anemia is also treated through somatic gene therapy. My body does not have some right genes. They can be corrected through gene therapy, और sickle cell anemia हम कल देखेंगे. Now there is germ cell gene therapy. This is for those cells in those genetic defects that have the potential to pass on to children. So in the germ cells, I have to give the gene therapy, right? Gene therapy can be used to fight cancer as well. That also we will see. This is called CAR T cell therapy. Okay. अब हम ये सारी चीजें देखते हैं कैसे होती है जीन थेरेपी. Virus containing the DNA is here. I add the human DNA containing the normal gene to the virus. What will form? Something like this. What is this? This is called chimeric DNA. What is a chimera? Chimera is a mythical or mythological. Exist नहीं करता actually में. It is a mythological animal. Okay, it is an animal that has a lion and has the wings of an eagle. Something like this. I'm not exactly sure. Like if you Google chimera, you'll see it is a lion. It has the wings of an eagle or a dragon, something like that. So it is a mix of two things, but it is not natural, right? That is why it is called chimera. ये chimera word का मतलब ही है an artificial mix of a lion and an eagle. So what is this chimeric DNA? This is the artificial mix of viral DNA and human DNA. जब मैंने वायरस के डीएनए को काट के बीच में अपना डीएनए इंसर्ट कर दिया तो क्या बन गया? काइमेरिक डीएनए. काइमेरिक डीएनए इज अ आर्टिफिशियल मिक्स ऑफ ह्यूमन एंड वायरल डीएनए. इट कैन ऑल्सो बी कॉल्ड रिकॉम्बिनेंट डीएनए. एक ही बात है. In fact, one of the applications of recombinant DNA technology is gene therapy. Okay. Now, chimeric DNA is inserted in the virus. अब आगे की कहानी तो आपको समझ आई गई होगी. वायरस जाएगा, सेल को इन्फेक्ट करेगा. When the virus goes inside, it will deliver the healthy gene to the nucleus, and the cell will then start producing the protein that was absent. है ना? बात वही है. This is the same thing we were doing for vaccine also. Same thing we are doing here. वहां हम एंटीजन प्रोड्यूस करा रहे थे, यहाँ हम कोई हेल्दी प्रोटीन प्रोड्यूस करा देते हैं जो हमारे पास ऑलरेडी नहीं है बिकॉज जीन इज मिसिंग. है ना? ओके, एक वीडियो देखते हैं. लेट्स सी हाउ इट वर्क्स.
In order for the muscles in our bodies to function, our cells need protein. In a healthy person, genes enable the body to produce the right proteins in the right amount. One protein that's very important for cell function is called survival of motor neuron, or SMN. In genetic disorders like type 1 spinal muscular atrophy, cells cannot produce enough healthy functioning SMN because they have a defective gene. In one of our clinical trials, researchers are using gene therapy to deliver the healthy SMN1 gene to a patient's cells. Researchers take viruses and modify them in the lab.
बेसिकली अभी तक इन्होंने क्या बताया कि स्पाइनल मस्कुलर एट्रोफी नाम की एक डिजीज है जिसमें एक एसएमएन जीन एब्सेंट होता है बच्चों में. अब उसको ये कैसे ठीक कर रहे हैं? दे आर यूजिंग वायरस एज वेक्टर एंड मॉडिफाइंग इट. आगे देखते हैं.
They only contain the SMN1 gene and do not cause harm to the patient. These viruses are loaded with the healthy SMN1 gene and injected into the body. When the material is injected, the viruses deliver the healthy SMN1 gene to the cells all over the body. Most importantly, they cross the blood-brain barrier to the motor neuron cells in the spinal cord. Once the virus has delivered the SMN1 gene to the cell, the virus disintegrates. The healthy SMN1 gene.
अब यहाँ क्या हुआ? यहाँ ये हुआ बेसिकली कि द वायरस इट हैज कम इनसाइड द न्यूक्लियस. इसने ये सेल मेम्ब्रेन पार करी, यहाँ आ गया. नाउ द वायरस इज हियर. इट हैज डिलीवर्ड द हेल्दी जीन इनसाइड द न्यूक्लियस. नाउ द हेल्दी जीन हैज बीन डिलीवर्ड. नाउ दिस न्यूरॉन or nerve cell will start forming the requisite protein.
Gene is taken into the cell but does not integrate with a native DNA. and no other genes are disturbed. Once the healthy gene is inside the cell, it can create normal functioning SMN protein. The goal is the good gene will produce enough good protein and the cell will function properly. Nerve cells that do not make good proteins will eventually die. When they die, the patient loses the ability to use his or her muscles. By delivering the healthy SMN1 gene to the cell before it dies, we can prevent the onset or stop the progression of SMA type 1 symptoms. For this reason, it's critical we identify and treat children as early as possible to give them the best possible outcome.
यहाँ इन्होंने क्या कहा? That it is possible that some cells don't have this SMN gene, so they will die very soon. So what do we need to do? We need to do this as quickly as possible and need to give as many rounds as possible that this gene gets delivered everywhere, right? So this is how gene therapy works. जितना यंग बेबी करेंगे, जितने यंग स्टेज पे हम ये काम कर लेंगे, उतना ज्यादा द जीन विल गो एंड गेट इनटू एवरी सेल, उतना ज्यादा द चांसेस आर दैट द सेल डेथ विल बी मिनिमल. जितना लेट करेंगे उतने ज्यादा सेल्स होंगे हमारे पास टारगेट करने के लिए, उतना मुश्किल होगा. So that is what they were saying, that it is critical that we do it in the early stage. I hope this is clear. If you look at the PDF, there is another video attached. आप वो भी देख सकते हो. You will get a better idea, but I'm not playing it here. Basic idea हमने बता दी. तो अब जीन थेरेपी हम क्यों यूज कर सकते हैं? मान लो कोई बैड जीन हुआ तो उसको मैं हेल्दी जीन से रिप्लेस कर दूं, ठीक है. Add a new gene to perform a function of a missing gene. एक तो है बैड जीन, ये जीन था बट इट्स नॉट प्रॉपर. एक है कि वो जीन ही मिसिंग है ही नहीं, उसको भी मैं न्यू जीन ऐड कर सकता हूं. तीसरा है कि देयर इज अ बैड जीन which in itself is doing no function. This gene in itself is doing no function, it is just causing a problem. It is not doing a function on its own. मतलब इसका जो प्रोटीन बनता है उसका नॉर्मली कोई काम नहीं है, ये सिर्फ एक प्रॉब्लम क्रिएट कर रहा है. जैसे मान लो कोई कैंसर की जीन है किसी में, तो आप उस जीन को भी सिर्फ इनएक्टिवेट कर सकते हो विदाउट एडिंग अ न्यू जीन. व्हाट विल यू डू? अब इसीलिए हमने म्यूटेशन तब पढ़ा था. इन द थर्ड केस, इफ देयर इज अ बैड जीन, लेट्स से somebody's DNA, I am not making it like this, ठीक है, I am making it straight. Somebody's DNA has a gene here that codes for a protein that causes cancer. So what will I do? I will take some enzyme or something and cut it here. When I cut it here, what will happen? It will form something like this, है ना. अब क्या होगा? हमने देखा था when these cuts happen, the cell does normal repair. सेल अपना नॉर्मली रिपेयर करने जाएगा, बट नॉर्मल रिपेयर के प्रोसेस में म्यूटेशन हो जाएगी, राइट. म्यूटेशंस हैपन इन द प्रोसेस ऑफ नॉर्मल रिपेयर. तो जब नॉर्मल रिपेयर होने लगेगा तो म्यूटेशन हो जाएगी. जब म्यूटेशन हो जाएगी तो जीन ओरिजिनल जो था दैट वाज अ कैंसर जीन. नाउ द कैंसर जीन गेट्स ऑल्टर्ड ड्यू टू द डिफेक्ट्स इन द नॉर्मल रिपेयर प्रोसेस. एंड दिस कैन ऑल्सो बी इंश्योर्ड कि नॉर्मल रिपेयर प्रोसेस बिल्कुल नॉर्मली ना चले, थोड़ा चेंज हो जाए. तो आप जैसे आपको बताया था, अगर इस जीन में एक भी जगह कहीं भी चेंज आ गया तो जो प्रोटीन बनना था वो डिफरेंट हो जाएगा. ओरिजिनल प्रोटीन वाज कैंसर कॉजिंग प्रोटीन, नाउ सम चेंज हैज इंट्रोड्यूस्ड इन दिस जीन कॉजिंग कैंसर. सो द प्रोटीन दैट इट वाज कोडिंग फॉर ओरिजिनली, द ओरिजिनल प्रोटीन वाज कैंसर कॉजिंग, बट दिस न्यू प्रोटीन is altered from that original protein, तो वो कैंसर कॉज नहीं करेगा. तो there are three things you can do with gene therapy. You can replace a bad gene with a good one, you can add a new gene, or you can turn off a problem gene. Now gene therapy can be done inside our body. Inside our body, how? Using virus. We have just seen in case of spinal muscular atrophy, SMA. Inside our body, virus will come and cause the gene therapy. It can also be done outside the body. Inside the body, when something is done, it is called in vivo. Inside the body, when something is done, it is called in vivo. When it is done outside, it is called in vitro. That is why you must have heard the term test tube baby. Test tube baby is used for the technology called in vitro fertilization. In the case of in vitro fertilization, fertilization between the sperm and the ovum is taking place inside a test tube. then the zygote, the first cell is put into the mother. So that is IVF, in vitro fertilization. In vivo is inside the body. Okay, that जो हमने देखा था gene therapy inside the body through the virus. How gene therapy can be done in vitro, in lab, outside the body, let us see now. In vitro gene therapy is done for a disease called adenosine deaminase deficiency leading to severe combined immunodeficiency. Please इसको याद करने की कोशिश मत करना. ADA skid is what you can remember. ADA skid. Although I'm not asking you to mug it up. I will just explain to you what is happening. ADA क्या है? ADA is adenosine deaminase. What it does is not important, but it is an enzyme. Adenosine deaminase, end में A's होता है तो mostly enzyme ही होते हैं. Adenosine deaminase is an enzyme. This is responsible for the normal functioning of T cells. T cells क्या करते हैं? एक तो helper और एक cell mediated immunity. T cells की normal functioning के लिए we need adenosine deaminase. अब कई लोगों में due to genetic defect what is happening? This ADA is absent. Adenosine deaminase is not there. So what happens to them is skid. If your ADA is not there, you will get skid. SCID stands for severe combined immunodeficiency. Immuno means immune, deficiency means absent. So you will have severe combined immunodeficiency if you don't have the enzyme for adenosine deaminase. Okay. अब मुझे क्या करना है? I need to give the T cells. The T cells need to be given the enzyme ADA. T cells now need कि उनको ADA दे दिया जाए. तो हम क्या करेंगे? हम पेशेंट के पहले ब्लड निकालेंगे. T cells are in the blood. ठीक है, हम ब्लड निकालेंगे. ब्लड से हम क्या करेंगे? T cells आइसोलेट करेंगे. We will isolate the T cells. Then we will put the T cells in a lab, in a culture like this. ऐसे रख देंगे T cells in a petri dish or a lab. तो पहले मैंने ब्लड निकाला, ब्लड से T cell निकाले, T cell को एक जगह रख दिया. ठीक है, फिर इन T cells को मैंने इन्फेक्ट कराया in the lab. In the lab, I am getting these T cells infected with a virus that is having the gene for ADA. अब ये मत समझना कि आर्टिफिशियली मैंने लैब को या सिंथेटिक मीडियम में मैंने वायरस को ग्रो करा दिया. मैंने लैब में, देखो इससे बहुत डाउट्स भी क्लियर हो जाएंगे. What I did is I took your blood. From the blood I got the T cells. T cells I put in a tray. Now this tray has only T cells. I have certain technology that will allow this T cell to remain living even outside the body. It will remain living even outside the body. Okay. When it remains living even outside the body, okay, in this case they are living cells. मैंने living cells को through a viral vector infect करा. I get them infected with a virus that carries the gene for ADA. Now virus will start from one, go to all. So virus will replicate. Now you might think that virus is being replicated artificially in lab. No, it is being done artificially, but it is not being done in an artificial medium. The question that we had discussed originally was, can virus be cultured in an artificial medium? The medium here is still T cells. I am making this virus infect every T cell. So virus is definitely making many copies of itself and infecting them. But it is not an artificial medium. It is being done artificially in a living medium. Living medium is T cells. Now when every T cell is infected with this virus, I have genetically edited this virus to carry this ADA gene. Okay. I have carried the ADA gene through the virus into every T cell. Then I will put these T cells, now they have the ADA gene, I will put these T cells back into the blood. What is the problem here? Of course, I can't take the whole blood out in one time. In one time, our body has about 5 liters of blood. At one time, at max, I can withdraw 500 ml. After that, it will become a problem. So I will extract 500 ml. I will give the blood, right? I will take the T cell. I will genetically give the ADA gene. Then ADA gene will lead to production of adenosine deaminase, and these T cells will become normal. So I have to now do this process over and over again. So multiple rounds are required. This is in vitro gene therapy. Gene therapy being done in a lab. We have done ADS kit, right? Gene therapy for adenosine deaminase deficiency, severe combined immunodeficiency. Correct? Okay. Now you are saying it is done in lab. In case the person is aged, like if I have this deficiency in my T cells, you will have to do it multiple times. You will have to take the T cell out. You will have to then do it, and not all my T cells can be taken out at once, right? So multiple rounds will have to be done till the time all my T cells get this, right? So I will continuously have to go through this throughout life. But if I do it in vivo at young age, then only one round will be sufficient. Okay, now let us understand CAR T cell therapy, chimeric antigen receptor T cell therapy. What is chimera? Chimera is when we combine two things that are not generally present together, right? Chimera. Okay. Chimeric antigen receptor T cell therapy is a way to modify my T cells to fight cancer. I am using gene therapy to modify my T cells to fight cancer. What am I doing? I am taking T cells from the patient's blood. मैंने blood में से T cells निकाल लिए. Now I have added a gene which was normally not there. I have added a gene called chimeric antigen receptor gene. I have taken the T cells out. In normal human T cells, this gene is not there, but I have added a new gene called chimeric antigen receptor gene. What does this chimeric antigen receptor gene do? This is a normal cell. This is a cancer cell. When a cancer cell is there, what is basically cancer? Cancer is कि ये cell uncontrolled तरीके से divide करता रहेगा. There will be no stopping it. Divide होता ही जाएगा ये. Correct? तो जब ये cancer cell होता है, cancer cell has certain receptors outside that a normal cell does not have. यहां पे वो नहीं है, but cancer cell में there are certain receptors outside. Okay. Now body normally does not have the ability to see that this is a foreign receptor. Our body normally does not have the ability to recognize these receptors. Because if it could recognize this as a foreign receptor, it would kill only the cancer cells. Normally when treatment is given for cancer, what happens is that we try and kill the cancer cell. But the problem is they are just like normal cells with two extra receptors. Cancer cells are just like the normal cells with two extra receptors. So if we give a drug or radiation to kill the cancer cell, normal cells also die. That is why cancer patients, when they are given therapy, their hair fall off, their weight reduces, because it's not possible for us to ensure that कि सिर्फ यही वाले cells मरे. If we are giving a cell killing substance, this will also be killed, this will also be killed, so it will lead to a problem, right? हमें कोई तरीका चाहिए कि सिर्फ यहाँ पे hit हो medicine, यहाँ hit ना हो. In this case, what can we do? We can take the T cells of our body. T cells are responsible for what? Cell mediated immunity. इनका normally काम भी disease cells को मारना है. So what will I do? I will take the T cell out. I will insert a new foreign gene in them called CAR, chimeric antigen receptor gene. This will lead to it forming a new receptor that can specifically recognize cancer cells. It will now generate a new receptor, a new protein that will allow it to specifically recognize cancer cells and not normal cells. Now this chimeric antigen receptor infused T cell, this T cell with this new CAR gene, when it comes and hits a cancer cell, it will be able to say that I want to kill this cell. When it comes and hits a normal cell, it will not kill that cell. Because from outside, I have introduced a chimeric antigen receptor. Why chimera? Because I am introducing artificial DNA. I am joining two parts of DNA not found together. So I will introduce chimeric antigen receptor inside the T cells it will now have new receptors. When it sees the cancer cell, it will kill it. When it sees normal cell, it will say, okay, fine, no problem. Okay, this is CAR T cell therapy. What have I done? I have used new genes. So it is a type of gene therapy. And what is it helping in? It is helping in treatment of cancer. Okay, now that you know genetic modification, you know ligation, you know sticky ends, you know restriction endonuclease, you know how genes are cut, how they are joined together. This thing is clear to you. Okay, all right. What are the benefits? It can help in curing certain cancers. The current method is chemotherapy to cure cancer, but chemotherapy has to happen multiple times, and it is the effect of killing normal cells also. So when you do chemotherapy, my hair will start falling off, my normal cells will also die, and I have to keep eating those medicines over and over again. In this case, I can do it only once. If I just do this gene therapy once with my T cells, they will now be able to kill all the cancer cells by themselves. They will understand, okay, this is cancer cell, this is normal cell. Okay, so the treatment is shorter, the recovery is faster. This is why CAR T cell therapy is now in news, because now it has been allowed. Okay, it has been allowed in India indigenously. It has been started to test. Indigenously, India is now testing CAR T cell therapy. Tests have started on patients. It has been allowed on patients now. That is why it's in the news. Okay, so I hope you've understood the concept. Okay, now what is the issues of gene therapy? Firstly, in certain cases, like we saw in ADA SCID, there have to be multiple rounds. Okay, there have to be multiple rounds in ADA SCID because every T cell has to be changed. And at one time, I can only withdraw 1/10 of the blood. So I will need minimum 10 rounds, right? Okay. The possible solution is you do it at an early stage. At an early stage, or the possible solution is you do it in vivo. Okay. Some side effects are that immune responses are possible. Okay. And it cannot be used to correct chromosomal disorders. I promise this is the last term for today. Gene therapy cannot be used to correct chromosomal disorders. What are chromosomal disorders we'll see, but I hope you have understood gene therapy. अगर नहीं समझ आया तो I'll make it very clear. कुछ genes normally present होती हैं. If those genes are absent or they are malfunctioning, I can insert new genes to make their function normal. Normal gene नहीं है, normal gene inactive है, non functional है, तो मैं बाहर से एक जीन लाके दे दूंगा. बाहर से वो जीन आएगी, वो अपना प्रोटीन बना लेगी. बाहर से वो जीन आके जब अपना प्रोटीन बना लेगी तो काम चल जाएगा. राइट, दिस इज जीन थेरेपी. एक एडीएस किड का एग्जांपल देखा जिसमें टी सेल्स में एक प्रोटीन नहीं था. टी सेल्स को बाहर निकाला, वायरस से इन्फेक्ट कराया, वायरस को मैंने सेफ बना दिया था जेनेटिक एडिटिंग से. वायरस में आई हैड पुट द एडीए जीन. वंस दैट इन्फेक्शन हैपेंस, नाउ ऑल सेल्स विल गेट द एडीए जीन एंड दे विल स्टार्ट फॉर्मिंग टी सेल्स विद एडीए. सो द डिजीज विल बी सॉल्व्ड. बट एक बारी में मैं सारे टी सेल्स बॉडी के निकाल नहीं सकता, सो आई विल हैव टू डू मल्टीपल राउंड्स. दूसरा देखा यूजिंग जीन थेरेपी टू फाइट कैंसर. I took the T cell out. I put a new receptor called chimeric antigen receptor. This chimeric antigen receptor can recognize cancer cells separately, does not see normal cells. Normal cells को नहीं मारता, cancer cells को मार देता है. So now I edited T cells in such a way that they could fight cancer. एक हमने देखा spinal muscular atrophy. उसमें क्या कर रहे थे? उसमें we were using virus inside the body. What will that be called? In vivo gene therapy. Okay, so I hope this much is clear to you. Issues हमने देख लिए. This is done, same slide है. Now last thing we are doing today is chromosomal disorders. What are chromosomal disorders and why they cannot be cured during meiosis? Okay, during meiosis, they cannot be cured with gene therapy. Gene therapy cannot be used to solve the issue of chromosomal disorder. Why? Why do chromosomal disorders happen? Let's say our body has 46 chromosomes. It has to do meiosis. Chromosomes exist as pairs. Okay? हमें पता है 2n exist करता है. So we have 23 pair of chromosomes. Okay? like this there are 23 pairs. Now in case of meiosis, what has to happen? First the cell will double its DNA. From 2n it will become 4n. So here it will start looking something like this. Okay, so now that it has doubled, the genetic material has doubled. Now it will be divided. It will be divided equally in 4 cells. All of them will have only n. Okay, this is what meiosis is. But मान लो meiosis में कोई defect हो जाए, when some defect happens in meiosis and parent, like the gametes that are formed, they don't have the normal copies, right? The division does not happen properly. तो एक क्रोमोसोम की एक्स्ट्रा कॉपी चली जाए कहां पे? That is a chromosomal defect. I'll just show you. I'm not sure how clear this is, but looks clear to me. Okay. So originally ये divide हुआ. Okay. This divided. यहाँ पे पहले double हो गया genetic material. अब split होना था. Like now after doubling, first doubling happened. Now it had to split equally in 4. But what happened? इन दोनों को तो normal एक एक क्रोमोसोम मिला. इसको दोनों मिल गए, इसको एक भी नहीं मिला. तो क्या हुआ? यहां पे दिस सेल हैज 24 क्रोमोसोम्स, दिस हैज 22, 23, 23. ये नॉर्मल है, ये नॉर्मल है, ये अब नॉर्मल है, इसमें एक्स्ट्रा कॉपी है, ये अब नॉर्मल है क्योंकि इसमें एक कॉपी कम है. तो ड्यूरिंग मियोसिस देयर इज सम डिफेक्ट व्हिच लीड्स टू नॉन इक्वल डिस्ट्रीब्यूशन ऑफ क्रोमोसोम्स. क्रोमोसोम्स इक्वली डिस्ट्रीब्यूट नहीं हुए. जब क्रोमोसोम्स इक्वली डिस्ट्रीब्यूट नहीं हुए तो क्या होगा? मियोसिस में हमने गमीट्स बना लिए, राइट? गमीट्स आर फॉर्म ड्यूरिंग मियोसिस. Now this is the egg. It has, the distribution was not equal, so it is normally supposed to have 23 pairs, or it's supposed to have 23 chromosomes, right? It is supposed to have 23 chromosomes, half haploid है ना, it is supposed to have n, 2n से it will have n haploid, so it's supposed to have 23 chromosomes, but it has 24. बिकॉज मियोसिस ढंग से नहीं हुआ तो इसे एक एक्स्ट्रा कॉपी मिल गया. कौन से क्रोमोसोम नंबर का? 21. क्रोमोसोम नंबर 21 का इट हैज एन एक्स्ट्रा कॉपी. सो इट हैज 1 क्रोमोसोम 1, 1 क्रोमोसोम 2, 1 क्रोमोसोम 3, बट एट क्रोमोसोम नंबर 2 इट हैज 2 कॉपीज. तो जब अब इसमें आके स्पर्म मिलेगा तो फाइनली व्हाट विल हैपन? क्रोमोसोम नंबर 1, 1 फ्रॉम द एग, 1 फ्रॉम द स्पर्म. क्रोमोसोम नंबर 2, 1 फ्रॉम द एग, 1 फ्रॉम द स्पर्म. बट जब हम क्रोमोसोम नंबर 21 पे जाएंगे so it will have 2 from the egg and 1 from the sperm, so the person will have 3 copies of chromosome 21 rather than the normal 2 copies. हर क्रोमोसोम की 2 कॉपीज हैं, एक मदर की एक फादर की, बट यहां मदर ने 21 नंबर क्रोमोसोम पे 2 कॉपीज दे दी और फादर ने एक दी, तो दिस बिकम्स chromosomal disorder. You have 3 copies of a particular chromosome. This condition is known as Down syndrome. ये सुना होगा आपने, बहुत ही कॉमन है. अनफॉर्चुनेटली a lot of people suffer from this. Patient has 3 copies of chromosome number 21. Why? Because there is a lack of proper meiosis. Meiosis did not happen properly. So there was chromosome number 21 had 3 copies. बस इतना याद रखना है, right? तो this cannot be cured by gene therapy क्योंकि पूरा क्रोमोसोम का प्रॉब्लम है. अगर एक जगह प्रॉब्लम होती तो ठीक था, राइट? 2 जगह प्रॉब्लम होती तो ठीक था, बट यहां तो पूरा एक क्रोमोसोम ही एक्स्ट्रा आ गया है. सो क्रोमोसोमल डिसऑर्डर्स कैन नॉट बी ट्रीटेड विद जीन थेरेपी बिकॉज पूरा एक इतना बड़ा सेगमेंट ऑफ डीएनए ही नया आ गया है, राइट? अगर एक छोटी एक जीन की प्रॉब्लम होती तो ठीक था. एवरी क्रोमोसोम हैज अबाउट 40-50 जीन्स, तो हम इसको नहीं ठीक कर पाएंगे. Chromosomal disorder, Down syndrome looks like this. They have this sort of face. You must be knowing someone who is suffering from this, right? They are always smiling. They have some heart defects. ये सारे features हैं Down syndrome के. There is growth failure, mental retardation, epicanthic fold of eyes. आपको ये याद नहीं रखने. I am just explaining. अगर आपको ये statement मिल जाए कि chromosomal disorders can be cured with gene therapy, it will be wrong. And why it will be wrong I have explained. Because chromosomal disorder means huge new copy of chromosome. जीन थेरेपी से हम एक आधी जीन ठीक कर सकते हैं, 200-300 जीन नहीं ठीक कर सकते.